Good morning, everyone. How's everybody today? Very well, thanks. Before we begin, I'd like to acknowledge that GEN-FISH, our universities, partners, and research benefit from and are situated on the traditional unceded territories and lands of Indigenous nations. We acknowledge the rights of the First Nation, Métis, and Inuit people of Turtle Island. We encourage all of you to reflect on where you are situated and its history, recognizing that participants will be in different territories. Now, my name is Joe Branjett, for those of you who don't know me. For those of you who do, I apologize. A couple of things. Welcome to AGM, GEN-FISH 2025, looking back and moving forward. This is Paige Bro. She is our research and communications coordinator and has been my right hand since May 2021. She's handled all the progress reports, ROC stuff, all the science stuff that none of which I understand. I deal with dollar signs and we're a great team. I have to say there's a quick story here. Margaret has pointed out and Paige has pointed out. So I have the vehicle with the 2 eDNA on the license plate. It's not eDNA. My late mother-in-law's name was Edna. So for those of you who think I'm really, really into this job, sorry to disappoint you. How was the breakfast? Was that good? Good. All right, excellent. Freck last night, show of hands, who attended? Who took the boat tour? Did you enjoy that? For arranging all of that last night, that was fantastic.
The underside of the Gordie Howe Bridge has holes. So
just like the Ambassador Bridge. Is that a design thing? Wow. For those of you staying at Canterbury, the accommodations are okay? All right. Thank you very much for being patient with us and understanding we had to flip this thing on its head to make it happen. Some housekeeping items. The washrooms, for those of you who haven't found them yet, the men's are just outside the door on the left. The women's are a little bit further down the hallway to the left. Women's are on the right side. There's a handicap washroom on the left. All right. We can click to the next slide, please, Paige. You'll notice in your lanyards, you have the agenda at a glance. And for those of you online, you wouldn't be joining without the agenda because it has the hyperlink to this meeting. So you can pull it up on your phone. You can flip it around for tomorrow and you see tomorrow's agenda at a glance. Just a quick highlight of what we'll be doing today. We are going to do our 5-minute presentations, which are recorded. We'll be playing those. And then we'll be going into our 15-minute presentations. We'll do 3 of them. We'll have a short break. We'll do 2 more, and then Margaret will talk about the HQP contributions to GEN-FISH. We'll break for lunch. We'll move into the toolkits in the afternoon, and we should be breaking about 4. Dinner is back here in this room at 6:30. Okay? So with that, I would like to invite Paige up. She will do a quick recap of all of the stuff that we've accomplished in this project. Thank you.
So yeah, before we get to the main events, I just wanted to kind of highlight all of the tremendous work and significant accomplishments that this research group has undertaken. Since GEN-FISH is kind of divided into activities, I think it can sometimes be easy to be siloed into individual sections and labs and theses and not be able to see the full picture. But Joe and I and other members of the support team have been in the unique position to get a broad fisheye lens view, if you will, of what this group has actually pulled off. So starting with activity 1, just recently, actually, Margaret Docker was saying after our last Research Oversight Committee meeting that if you go back and look at the GEN-FISH proposal, what was actually proposed was close to insanity. But as this saying goes, you shoot for the moon, you tend to land among the stars. And in this case, the stars are 183 CON assays and 175 Site B assays that have been designed during GEN-FISH. This represents over 80% of all freshwater fish species in Canada. And not only that, but 5 taxonomic and 9 regional open array chips have been designed and validated, none of which were part of the original proposal, but all were added based on feedback from our co-producers. So this collection of assays has broken down at quite a large scale, significant barriers that normally would prevent decision makers and practitioners from being able to use eDNA technology at all. Then we have activity 2. What we've been calling the 500 water body survey is in reality close to 1,500 sites sampled across every province and territory in Canada. The deployment of students, researchers, and community groups to collect eDNA over the last 5 field seasons has culminated in the first and largest pan-national eDNA survey of its kind. As Matthew Yates can attest to, the data from this survey can and will be used for broad-scale analyses for likely years to come. And what began as 9 case studies with partner groups has doubled, with 10 already complete and 7 wrapping up this year. Activity 3 has also greatly adapted from its initial vision of developing one universal stress response transcriptional profiling chip when it became clear that the candidate genes were not universally expressed across taxonomic groups. The Activity 3 team pivoted to no fewer than 20 iterative order-specific STP chips. These chips can address and investigate stressors ranging from transport stress to circadian rhythm to osmoregulatory stress across 6 orders of fishes. On top of that, there have been multiple chips developed that were not part of the proposal, such as the American eel chip and a chip that can target genes expressed in mucus, which allows for truly non-lethal and minimally invasive sample collection. So from the outside, and I can say this personally, this process of chip design and testing has appeared so streamlined, it almost hides the tremendous amount of time and dedicated work behind each version of each STP chip. And all the efforts of Activity 3 were put into practice in Activity 4, where the STP chips were employed in addressing the concerns and questions of resource managers across the country. So salinity tolerance, thermal stress, predator stresses, water quality stressors were examined in 16 case studies across 12 species. The entire transcriptional profiling side of GEN-FISH has expanded what I think is a fairly new field of applied omics in fisheries management. I'm sure all of these studies will be considered foundational for future applications. And all of these technical and analytical achievements would be released into a void of a knowledge gap were it not for the efforts of our Activity 5 members. So thanks to these folks, we have feedback about specific concerns that real people have about using eDNA transcriptomics, concerns like the collection of human DNA and what informed consent would look like, security of collected data and samples, and the consequences when commercial interests are placed above environmental stewardship. Paired with studies like Tongji Li's economics game, if you were here in 2022, results from 1,000 surveyed anglers and the MCDA tool currently being tested on the website. This means that not only have we advanced these technical fields, but we have tools and resources on how to put them to the most effective use. But here's some really exciting numbers. And it comes down to the real drivers behind GEN-FISH, the HQP. We've been using the acronym for quite a while. I think it can be easy to forget that these are our highly qualified personnel, and this project has been run by 184 of them. GEN-FISH has been the vehicle by which 121 students have gained expertise and experience enough to earn 41 undergraduate, 52 master's, and 28 PhD degrees. And much of our project continuity has been supported by 46 lab technicians and research assistants and 17 postdocs. Our members have written and contributed to 67 and counting peer-reviewed publications. This is by all accounts a very conservative estimate, and the manuscripts won't be slowing down anytime soon. More so, our researchers have been recognized with 10 awards at local, national, and international levels, and GEN-FISH research has been showcased in at least 77 conference presentations since 2020. There's been a huge number of people working quietly behind the scenes to keep this project running as well as we can. Joe, our long-suffering project manager, who we entirely owe this whole AGM to. There's, of course, Sarah Jameson, who launched so much of what we take for granted now, and I believe we honor her through the completion of this project. There are all of the lab technicians who have supplied their expertise and many more in each lab. It goes without saying that we would have been entirely and completely lost without you. So there's, of course, a bit more work to be done. Certainly feels that way. But I hope over the course of the next 2 days, today and tomorrow, you take the time to congratulate one another, all of your efforts, all of your successes, and in completing this large-scale applied research project. So thank you, and now I'm going to queue up our first 5-minute presentation.
Couple of rules for this, well, actually one main rule for this weekend. Have fun. There's no ROC. If things don't work out, we're just going to enjoy this moment. This has been a 5.5-year ride. We are just reflecting and enjoying what we've accomplished. So just enjoy this. For all of the people doing the 15-minute presentations, don't be nervous. Just have fun. Thanks.
Hello everyone. Today I'm going to briefly present our research on comparing automated offshore eDNA sampling and gill netting to detect fish assemblages. This research has been conducted in collaboration with the Saugeen Ojibwe Nation, in particular the Environment Office. I want to acknowledge the traditional territory of the Anishinaabe Nation, the people of the Three Fires known as Ojibwe, Ottawa, and Potawatomi Nations, and further give thanks to the Chippewas of Saugeen and Chippewas of Nawash, now known as the Saugeen Ojibwe Nation, as they are the traditional keepers of this land. Further, I want to thank Kathleen Ryan and Son Harvesters for making the autosampler deployments happen and for their help troubleshooting. So an eDNA autosampler is a device that is programmed to collect water and pull it through a filter in a filter housing, then run preservative through the filter, and this is done at a preset time interval. The reason for deploying this autosampler is to survey the fish assemblage around a proposed pump storage site that would require dredging in the nearshore sediment of Georgian Bay for its construction, then once built would intake water up the escarpment and then later release it back into the bay. The potential benefits of an autosampler would be reduced human sampling over this military hazard zone, its ability to collect eDNA at night, and it might be a more sensitive fish detection method than gill netting. Near Meaford, Ontario, we had 4 autosampler deployment periods, 2 in a reference zone and 2 in hazard zone 2. North-South consultants set and retrieved gill nets over this period, allowing for comparison of fish assemblages between sampling methods. On the map, the triangles are autosampler locations and circles are gill net locations. We found that eDNA metabarcoding detected 17 species, whereas gill nets detected 20 species. Some of the main assemblage differences are eDNA detected higher proportions of small-bodied fishes, and gill nets tended to catch more medium-bodied fishes. These trends could be due to smaller fishes producing more DNA per unit biomass and general higher abundance, or smaller mesh nets being more easily spotted and avoided by fishes. I also made species accumulation curves with Chow-2 richness estimates to get an idea of sampling effort required to detect fish assemblages using each sampling method. The red curves are eDNA richness per sampling unit, and the blue are gill net. For each curve, the points are observed richness, and the left of the points are rarefied richnesses, and to the right of the points are extrapolated richnesses. So eDNA curves from deployments 2 and 4 indicate at least 25 filters are needed to sufficiently detect the local fish assemblage. Gillnet curves from deployment 1 and deployment 2 show at least 50 gangs would be needed to detect the fish assemblage using gillnets. However, in curves for deployments 2 and 4, we see that the eDNA, so the red curve, as the red curves plateau, they are unable to meet the observed richnesses from gill netting. This indicates that eDNA sampling could be insufficient and not as effective as gill netting. Although gill netting appears to be more effective, if we increase the number of filters that were collected in a deployment period, we might see richness estimates comparable to gill nets. However, increasing frequency of filtrations would require more frequent deployments or more auto-samplers to be deployed. This is both costly and labor-intensive. It's also difficult to estimate the rate of eDNA degradation, especially when some filters remain in the water for up to 13 days following filtration. I plan on looking at positive controls for indication of degradation. Lastly, we were able to detect some species using eDNA that were not caught in gill nets, and we also observed differences in size classes between fishes detected by both methods. So this supports the recommendation to implement multiple year types to survey fish assemblages whenever possible. Thank you, and if you have any questions, please reach out to me via this email.
Hi everyone, I'm Patricia Boyer. I am a master's student in the Heath Lab at the University of Windsor, and today I will be sharing with you a brief overview of my master's project, which is focused on gene transcription variation in response to environmental stress gradients in Canadian freshwater fishes. And for this final GEN-FISH AGM, I'll also highlight progress on 2 GEN-FISH STP chips and share an example of their application. Environmental stressors pose great threats to aquatic ecosystems around the world. They can come in many forms, including pollution, temperature, acidification, and many others, and they are often the result of anthropogenic activity. It is important to understand, however, that environmental stressors are not uniformly distributed across all waterways, but rather they tend to occur over a gradient. That is, with spatial variation at higher and lower levels, and we typically see multiple stressors occurring together, as opposed to just a single stressor in an ecosystem. And so in recognition of the salient issue worldwide, for my master's project, we set out to test for adaptive phenotypic plasticity and or flexibility among wild fish populations in response to both thermal and nutrient pollution environmental stress gradients by quantifying gene transcription patterns. We began by taking advantage of the Bay of Quinty's natural thermal gradient, collecting yellow perch from the upper to the lower bays in the summers of 2022 and 2023. During the summer of 2022, yellow perch, tessellated darter, and white sucker were sampled following a forested to agricultural nutrient pollution gradient along Lac St. Pierre, where temperatures ranged from about 17 to 29 degrees. And finally, working in collaboration with the Ontario MNRF, we were able to collect yellow perch across a latitudinal gradient ranging from about 43.4 up to 49.5 degrees north during the summer of 2023. And so since my master's project encompasses quite a wide range of temperatures, spanning approximately 15 to 29 degrees Celsius, and also different nutrient pollution loads, to effectively assess whether the fish in the sampled populations were stressed or perhaps exhibiting signs of local adaptation over a gradient, there was a need for reliable assays. And so I began by designing and optimizing 2 order-level open-array stress transcriptional profiling chips. or STP chips, one for perciforms and one for cypriniforms, that would be fit for not only the target species in my project, but also other GEN-FISH research projects. And this included genes from several gene categories that are indicative of different environmental stress, as shown on the screen. Now, as part of the optimization process, cDNA from relevant and available species and tissue types was loaded onto the STP chips and run on an open array machine. Overall, the chips performed very well and amplified cDNA for most genes across different species within the cypriniforms and the perciforms orders. And especially our target species, whitesucker, yellow perch, and tessellated darter, showed amplification across many genes, and importantly, genes from every functional category were represented. But I thought I'd share with you an example of how we've applied the perciforms STP chip and some preliminary findings from the yellow perch that were sampled along a latitudinal gradient. Here I've constructed a heat map showing the relative gene transcription of the yellow perch collected from several lakes, ranging from Conestoga, our southernmost lake, up to Rugby, our northernmost lake. Now, at first glance, these preliminary findings do not necessarily highlight a stress response, but rather we see very low levels of gene transcription levels across several stress-related genes, and this is likely because most of the lakes were quite cool. However, I'd like to point out that two of these lakes, Rugby and White Otter, share very similar gene transcription profiles. And interestingly, these two lakes are in relatively close geographic proximity to one another. Now, this could potentially indicate local adaptation or perhaps shared environmental influences between these two lakes, but further analyses will be required to draw such conclusions. In this ongoing project, as we continue to broaden our analyses to other sites across Ontario and Quebec, we aim to establish and quantify the threshold at which environmental gradients will induce a measurable stress response, which will enable us to predict the point at which stressors negatively impact fitness in natural ecosystems. And finally, the development of both the cypriniforms and the perciforms STP chips will serve to help monitor fish health in a cost-efficient manner, and it will help us to mitigate potential threats to ecosystems. In closing, I wish to thank all those who have helped with this project, and thank you, GEN-FISH, for watching my presentation.
Following their initial establishment in the Great Lakes in 1922, rainbow smelt have caused a number of issues within the system. They contain the enzyme thiaminase, which is responsible for causing thiamine deficiency in predators of the smelt, such as trout and salmon, and they also outcompete native prey fish. They've become one of the most abundant fish in the Great Lakes. Despite this, little is known about their origins in the system or how they became introduced. It's known that smelt from the Green Lake Hatchery in Maine were introduced to Lake Michigan in 1912, and they spread from there to Lake Superior and Lake Huron. However, smelt were found in the easternmost Great Lake, Lake Ontario, prior to being found in Lake Erie, and this is inconsistent with the idea of a radiation outwards from a stocking in Lake Michigan. There have been three major hypotheses that have been proposed to explain this discrepancy. First of all, it's possible that smelt simply weren't detected in Lake Erie for 6 years while they spread to Lake Ontario. This is unlikely given how much fishing pressure was on Lake Erie at the time and how populated the shoreline was. Alternatively, a second anthropogenic introduction could have occurred in the vicinity of Lake Ontario. This could have been smelt from the same source, the Green Lake Hatchery, or from the other hatchery that existed at that time, the Cold Spring Harbor Hatchery, which cultured saltwater smelt from New York State's Atlantic coast. The third hypothesis is that smelt from the St. Lawrence River moved upstream into Lake Ontario from their native range in that river, and that explains the premature sightings of rainbow smelt in Lake Ontario. Smelt vary significantly in their thiaminase activity, their size, their diet, and other life history traits across their geographic range. And so knowing where smelt came from and what their population structure in the Great Lakes is important for managing this species and predicting changes in the population that will occur over time. One way that we can answer these questions is through the use of genetic tools. There are two major mitochondrial lineages of rainbow smelt, and the Green Lake hatchery cultured the Atlantic lineage, whereas the Cold Spring Harbor hatchery cultured the Acadian lineage. And so by examining SNPs associated with these different lineages, we can determine the origins of the smelt in the Great Lakes region. In our study, we examined smelt from Lake Ontario, Lake Erie, and Lake Superior, and as well as some lakes in northern Ontario. And we used restriction fragment length polymorphisms and Sanger sequencing to identify which clades were present. And what we found was that both clades were present across the Great Lakes region. And this suggests that two introductions at least occurred from two different sources, the Cold Spring Harbor hatchery and the Green Lake hatchery. In addition, we found that none of the smelt that we sequenced showed high affinity to the St. Lawrence River smelt, which suggests that no native smelt entered through that pathway. And finally, we found that the frequencies of the Atlantic clade and the Acadian clade smelt were fairly homogeneous across all the lakes studied. And this suggests that there's high connectivity in this environment and that smelt have dispersed across the Great Lakes since their introduction. And this indicates that the phenotypic differences that we see across the Great Lakes are likely not due to differences in ancestry, but are rather due to factors such as the environment in those specific sampling locations.
Okay. Good morning, everybody. My name is Andrew McDermott-Fouts, and I studied at UNBC in Dr. Shrimpton's lab. And my master's project was all about piloting eDNA, both metabarcoding and qPCR, in the Upper Peace watershed. And really, in this 5-minute presentation, all I got to have time for is to show you this picture as to where it is. But one of the most defining features of the Upper Peace is the fact that it's dammed 3 times now, and it's created the largest water body in largest freshwater body in British Columbia, which is the Williston Reservoir. And so that was my study area. But today I want to talk about some of the challenges that I had in developing qPCR assays. And particularly for two salmonid species that are really closely related. So in BC, we don't have a ton of species diversity when it comes to freshwater fish, and a lot of those species are very closely related to one another, and the salmonids are really characteristic of that. But two of the salmonid species that were coexisting in the Williston Reservoir were bull trout and lake trout. And I know some of you in the room know a little bit about bull trout and lake trout, but bull trout's distribution is more western. It's really from the foothills of Alberta to the coast and then up into the Arctic and as far down as the United States. Lake trout, on the other hand, are pretty widely distributed across Canada. So there is some overlap in British Columbia. And they're both char species. So they're both in the same genus. And as far as their mitochondrial DNA, it's very similar. So this was a challenge for developing an assay for both of those species. Now, there were previously published assays for both species. And of course, because the distribution overlaps in British Columbia or in the West with bull trout, their assays were quite effective in differentiating between those two species. Meanwhile, the lake trout assay was developed out here in Eastern Canada, where you don't have bull trout. So unfortunately, it was a complete match for bull trout DNA. And I had to develop a novel assay that would do the difficult task of parsing those two species. So to develop a species-specific assay for lake trout, I had to design primers and probes that did quite frankly, a number of things, but the most important of which was that they didn't amplify bull trout DNA. And so the lake trout Site B assay that I finally or eventually got to is along the top of that table. And then the bull trout sequence or complementary sequence below it is shown with the mismatches in colors. And I'll get to the colors in a second. But generally speaking, the more mismatches you have between your assay and the non-target sequence, the better. And so I was looking for a different, or for the best gene to get that sequence divergence between the two species. The best one was the Site B gene, but even then I was only able to cram 6 mismatches total into all the sequences. So because I only had 6, I had to really optimize and make best use of those for specificity. So the first part was to position them in the places where they had the most influence, and that was in the primers primarily because they have more of an influence on specificity than the probes, and specifically in the 3' end of those primers. So I got really lucky with the rear primer because I was able to jam these 2 really closely spaced primers right at the end. So that was good. And in the forward primer, I didn't get as lucky because the 2 mismatches were a little bit further apart. But I was able to get one right on the 3' end, and that one was pretty key. And that is because it is a purine to purine mismatch, which has been described as a critical mismatch. And that is because it reduces the amplification efficiency more than other mismatches. And that's why it's red versus blue. And so by trying a number of different combinations, I was able to come up with an assay that we validated in the lab in vitro. And then we took it up to the Wilson Reservoir. And unfortunately, we sampled in an area that we didn't have any lake trout, so we weren't able to fully validate that assay. But it still can be done. We just got to take it to a water body or take some tests from a water body where we know there's lots of lake trout and do a little bit more testing on it, and we'll get there. But this assay, I think, has a lot of potential for monitoring lake trout in the West because we have to deal with those closely related species, particularly bull trout. And most of all, I think it's a really interesting case study for eDNA assay development when you have closely related species that cohabitate in or have similar ranges or overlapping ranges, as we do in many parts of Canada and specifically in British Columbia. And with that, I just have a few people to thank. And I don't know if there's question periods or I don't know. I suppose we have time now that we don't have. No hard questions. I've already defended, so. No.
Had my chance to give.
Yeah, you did. Well, if there's none, I can slink off the stage. All right, thank you.
Presentation winners is
Are we done with the 5-minute presentations?
yes, you're up.
Okay, awesome. I'm just going to start my timer so that I can keep track as well. Great. Thank you for having me. I'm sad to not be there in person. Again, I'm tuning in from Girdwood. It's been challenging to work so far from the lab, but it's nice to see everybody, even if it's just on a screen. So happy to be here. So I'd like to start by expressing gratitude to the University of Windsor and the GEN-FISH project, to those who designed the open array chips that I used, and also to the Gatineau Fisheries Authority for funding and allowing me to put these tools to use on their precious salmon. So I use these tools to ask the question, is smolt quality limiting the sustainability of a small genetically unique sockeye salmon population in the Skeena River watershed in British Columbia? Next slide. The Skeena watershed is shown in red on the left and is the second largest watershed in BC, second to the Fraser. I'd like to acknowledge the 5 nations that call the Skeena home: the Tsimshian, the Gitxsan, Gitineau, the Wet'suwet'en, and Lake Babine Nation. And these areas support several distinct populations of salmon. The star marks my study lake, Gitineau, which is also referred to as Kitwankul or Kitwanga Lake. You might have seen it if you were driving up towards Alaska from the Terrace area, because it's kind of where the 2 highways intersect. Next slide, please. Specifically, Gitineau Lake sockeye salmon are recognized as a significant conservation unit under Canada's federal wild salmon policy because of their small population size and genetic uniqueness from other sockeye stocks in the Skeena. Next slide. Gitineau Lake is a small but productive lake that has historically had a consistent run of sockeye salmon. But the run has seen considerable declines, like we've seen in many other places over the last few decades. And this is escapement since 2003. And if you look at the 2024, we only have 2,400 fish returning. So since about the 1970s, there's been no food, social, ceremonial, or economic harvest of sockeye for the Gitineau First Nation. So cause for concern. Next slide. On the left is a map of Gitineau Lake, a very shallow eutrophic lake, so sort of unique in terms of sockeye lakes in the area, in the Skeena. It's warm in the summer and deep layers become anoxic. So on the right-hand side, we can see data showing what happens throughout the summer in the water column, and we can see an increase in surface temperatures on the top right graph, and on the bottom right we can see a decrease in dissolved oxygen from the bottom. This creates what we've defined as a habitat squeeze. So as you can imagine, as things warm up from the top and we lose our dissolved oxygen from the bottom, there kind of becomes this narrow band that is suitable for fish to exist. And this sort of begs the question when we start looking at, okay, there's declines, what's happening in the lake, what's happening to juvenile sockeye in the lake through those summer months, and what might be the product of living in such an environment. Next slide. So after meeting with GFA, we defined study objectives to include a better understanding of this small significant conservation unit, and to better understand why sockeye are declining. Is it possibly related to some aspect of juvenile rearing? And to better understand the effect of lake rearing on smolt production specifically. And to answer these questions, we wanted to test the GEN-FISH OA chips as part of the Fish Health Toolkit. Next slide. To answer these questions, I compared Gitineau Lake smolts, which travel along the green route, to Babine Lake smolts, which is the blue, and they are the Babine Lake population is the largest producer of the Skeena, so a very robust population and a good one for us to compare Gitineau smolts to if we want to see how Gitineau smolts are doing. Next slide. So in order to answer these questions, I employed 3 main methods. And first we looked at size and age because we know that that is an important physiological parameter for smolts. And then I was also able to use transcriptional profiling to look at expression of several different genes, including some that are related to things like stress, osmoregulation, freshwater and saltwater ion regulation, hypoxia, and more. And then finally, I also used the NKA marker, which is a common marker that's been used for decades to determine smolt readiness for the ocean, and kind of loops us back to biochemistry. Sometimes post-translational effects can alter the expression of a gene in the end. So when we ask how is an animal performing, this kind of helps us ground truth with a physiological component. Slide 9. Sorry, next slide. Sampling took place at both the Kitwanga and Babine smolt fences. So these are long existing fences that they put in each spring, and they've collected, both nations have collected a lot of data there. It also gave us a unique opportunity to train GFA staff on how to use different kinds of tools. So they were able to learn about RNA extraction and sampling, as well as NK and gill sampling. So after taking samples from the field, we extracted the RNA in the Shrimpton lab, and then samples were sent to GLEAR. Next slide. Oh, okay, thank you. Once at GLEAR, samples were run on 2 open array chips. So I used a stress transcriptional profile chip for liver tissue and an osmoregulatory stress transcriptional profile, say that 10 times fast, for gill tissue. And of course, part of activity 4, fish health toolkits and transcriptional profiling. Next slide. Aging scale showed that both populations, Gitnyao and Babine Lake, migrated predominantly as 1-year-olds. So that gives us a good idea of how direct our comparison is here. And we found that there was a significant difference between Gitnyao and Babine smolts in terms of length, mass, and condition factor. So length is A, B is mass, C is condition factor. And this is not really a surprise because we know that Gitnyao smolts are some of the largest in the province. And this is a picture of them on the cutting board. And I don't have, unfortunately, there was nothing there to truly measure scale, but these fish get up to about 10 to 12 grams. So they're pretty large fish. Next slide, please. The OSTP OA chips results showed that not many genes differed. So I ran a PCA on all of the genes on that chip. So it was something around like 19 out of the 28 that worked. There was no difference in PC scores with loadings for saltwater ion regulatory genes, freshwater ion regulatory genes, and immune function, which is in plot A. PC1 and PC2 included those genes. In PC3, we didn't see a difference in stress functional genes. However, with PC4, we see a difference in growth and cortisol breakdown. In the bottom right, I show GHR and HSD11 beta. And these are the genes that loaded into that PC4, showing the difference associated with growth and cortisol regulation. So Gitnyao fish are perhaps not actively growing as fast as Babine at the time of smolting, but we do know that they are in fact bigger, so perhaps a growth spurt is happening before they are leaving the lake. Next slide. The STP-OA chip, which I used for liver samples, had, again, that 112 genes. Not all worked, but over half, I think about 57 worked, which was really great. I ran a PCA for 8 functional groups instead of across the whole lot of all the genes. And we saw, so some of those groups include stress, detoxification, endocrine disruption, circadian rhythm, hypoxia, which, again, a great chip for an environment like Gitnyao Lake to put to the test. So I ran the PCA for 8 functional groups and then looked for a significant difference between Gitnyao and Babine fish. Principal components for stress response, you can see the ones that are starred on the right in the red table. Stress response, endocrine disruption, circadian rhythm, and growth and metabolism were found to have a significant difference. But again, not really many genes differed. Next slide. Significant differences in growth and metabolism rated genes were observed for both gill and liver tissue, which is interesting. And this is a good example to compare to the OSTP chip. Within the growth functional group, PC2 was significantly different. GHR also supports our gill data because we saw higher levels in babbing fish. We also saw differences in IGF-2 with babbing fish expressing this gene at a higher level. And this suggests that babbing smolts are growing faster when they leave the lake, although they are much smaller. Next slide, please. We also saw differences in circadian rhythm, but there's a link to metabolic rate here for these genes. And if babbing fish are growing faster, that's not surprising. They might be migrating later. Next slide. And finally, when we compare to NKA activity, that physiological marker that is a strong indicator of whether a fish is ready to go to saltwater, we found no difference between populations. So this gives us a few variables to suggest that Gitniau Lake are competent and ready to go to the ocean, just like smolts from babbing. Next slide. Results indicate response to the habitat squeeze that we saw. So that increase in temperature and loss of dissolved oxygen is not overwhelming and doesn't appear to be a bottleneck for Gitniau sockeye salmon viability. Next slide. Just a little peek on some of the other work that's being done. So we have a good idea about what's happening with the smolts, but we also are investigating across that habitat squeeze gene expression changes before, during, and after. Most genes do change over time in our preliminary analysis. Upregulation is shown in green, downregulation in blue. But we're asking the question, does this suggest poor growth conditions for juvenile sockeye salmon in their first year? So those results are yet to come. Last slide. And with that, thank you to everybody. If you'd like my contact information, you can just click on that or take a picture of the QR code if you have any more questions. Thank you.
Thanks so much. Any quick questions?
Carolyn, I really enjoyed your presentation. Thank you very much. It's really clearly laid out and highly digestible. I was wondering if whether or not any of the nations you worked with have their own reasonings behind why they see this kind of a statement or difference between the two lakes. I'm just wondering what, your co-producers also thought about the reasons behind it and what they also think about the data that you showed them, presuming that they've seen the data already.
Sure. So I'm just going to paraphrase to make sure that I understand correctly. You're asking if the nation that I'm working with has any stipulations or ideas about why the population is decreasing?
Yes.
Okay, so that was the first part, and then we can move to the second part. So there's a lot of different things going on. They're also wondering about predation because there are also trout in the lake that eat a lot of the eggs. So that's something that they're thinking about. They've also done some research on how productive the lake actually is in terms of nutrient loading and things like that. And then another interesting piece is beaver dams. So there were some questions around they have to remove a lot of beaver dams every year. So is that causing holding fish back or in some way they're just not able to get to the lake. And then also chapter 1 of my thesis, we looked at there's a lot of residual, smaller, almost kokanee sockeye that are showing up on the spawning beds, and they're wondering if maybe more fish are staying and not going to the ocean at all. So shifting to a kokanee lifestyle. And we did see that there were a lot of, using otolith microchemistry, there were a lot of first generation residual fish. So maternal origin mom did go to the ocean, but they are staying in the lake.
That's so neat.
Yeah, super neat. There was a second half to that question about the data. I'm sorry, I've forgotten.
Yeah, no, it's just when you co-produce the results and you show the results, were they, your First Nations, how they interpreted it? They're like, you know what, yeah, we knew this all along, or how was it working with the First Nations to present this type of data?
Yeah, so I mean, we're probably due to have another conversation with them because we've it's been a little bit to kind of learn how to work the gene expression data in a way that we could tell a story with it. So we're due to have another conversation with them. But I think them knowing that, like, hey, your smolts seem fairly robust in comparison to these baboon fish has been, I guess, a little bit of a sense of relief, because it's like, okay, that's not the smoking gun.
That's great. Thank you so much.
Thanks.
Thanks again, Carolyn.
Okay, everyone. I'll start my timer. I've picked up a bad habit of going over time. I'm told it's like really relaxed here. So, hey, so I work with bull trout, and I work primarily with them because DFO is interested in them, specifically our collaborators, and so are a conservation group that I work with, so an NGO. So bull trout, they're distributed throughout Western North America, from Idaho all the way to the Northwest Territories. And what's cool about them is that they are cold water stenotherms, so they like a narrow range of cold water temperatures. So some of the projections, however, like many of the freshwater fish, there's a lot that threatens them. There's competition with other salmonids, fishing pressures. But with these fish in particular, what we're worried about is increasing temperature with climate change. So these are projections for the Northwest Territories. So by 2080, we're expecting a decrease in about 40% of suitable thermal habitat for these animals. So we used to not know a ton about the physiology of bull trout, but between me and Carol Beth, we filled out some of these blanks. So their preferred temperature range, at least in the lab studies that we conducted, is between 9 degrees and 14 degrees. And beyond that, we see a decrease in their physiological functions. So decrease in growth, decrease in aerobic scope. So they kind of don't do as well at 15 degrees and higher. And this is from a different study, and I'm happy to talk about this over the break. It's one of my favorite studies, actually. So I fit segmented models on these gene expressions over a range of temperatures. And we're seeing, this is just for gill, but we see a consistent breakpoint at about 14 degrees, in all the different tissues I've tested. So I did this in gill, liver, and muscle, and Carol also sees the same breakpoint as well in a different life stage. But what we want to know is whether mucus is a viable matrix to see these temperature thresholds in bull trout. So the mucus is a protective barrier between the animal and its external environment. It acts as a physical and chemical barrier. So some of the more common roles that you'll see in the literature is protection against pathogens, pollutants, and abrasions, but it also acts to regulate ion concentration, facilitates locomotion. Parental care is a cool one that I've read in the literature, and also chemical communication. So there's pheromones in there and stuff. So it's released primarily from goblet cells. So I'm focusing on epidermal mucus, so skin mucus. So it's released from goblet cells, mostly made out of water and glycoproteins. But what I'm interested in here is the RNA packages that are in the mucus. So they're packaged in microvesicles, or they're just released when cells break open. So this mucus, from previous studies, we know that the composition of the, the composition and the RNA expression does change with developmental stage. It can change with sex and environmental conditions. So this suggests that this matrix might be useful in determining whether fish are experiencing thermal stress. Also today, I'm focused between sexes. So I was able to sample male and female bull trout. And often in the literature for salmonids, we will see that females are having a harder time to overcome stressors in the wild. A lot of this literature comes from Pacific salmon primarily, but we see a lot of these like higher cortisol levels in females across teleos and a slower recovery from stress as well. So what I focused on is on tissue-specific transcriptional responses, and I'm expecting the mucus and gill to be most similar here. This is based on previous studies I've conducted with lake trout, but also these are the two external facing tissues that I've sampled. This was a misguided hypothesis, but muscle and liver, I thought they'd be doing the same thing, but they're very different tissues, and I knew that. But differences in thermal tolerance between sexes. So I'm showing an increase here, but you can also imagine a decrease. But I'm expecting females to show a shift in their transcriptional response at an earlier or at a lower temperature compared to males. Okay, so for this, we collected gametes, from the traditional territories of the people of the Treaty 7 region in southern Alberta. So this is in Smith Dorian Creek in Kananaskis. So these gametes, so eggs and sperm were brought back to the Fisheries Institute, the Fisheries and Oceans Fishery Institute in Winnipeg, where we raised the eggs at 4 degrees. And when they were fried, they were held at 10 degrees constant for 2 years. So for the transcriptional profiling, I sampled 12 to 15 fish after they were exposed to these 4 different temperatures for 4 weeks. So the 9 and 12 degrees here, those are 2 temperatures that they like. They prefer those temperatures and they tend to do well. 15 and 18 degrees here, that's where we see generally from previous studies, the onset of a cellular stress response and a decrease in physiological function. So that's why those 4 temperatures. Epidermal mucus swabs were collected. So these are nice because anyone can just pop in and help me sample. All you need to do is swab these fish, just give them a little scrub. Gill, liver, and muscle dissection. So epidermal mucus, non-lethal, but they were lethally sampled so that I can compare to the other tissue. So gill, liver, and muscle dissections. Something I'm not showing here is that I had 10 additional fish that we just swabbed and popped them back into their tanks. And I just wanted to show that there's no mortality. So the fish do fine after I give them a little swabbing. So everyone's seen this before. The chip that I used was the version 3 STP chip, so the stress transcriptional profiling chip. So there's still 56 genes on there with the different broad functional groups. And we're lucky enough that we have the Quant Studio 12 in Ken's lab, so I can run these chips. So this looks a little messy, but this is to show that in the mucus, so the mucus doesn't do a great job at separating between males and females, but that's not what we wanted it for. We want to know whether these fish are stressed or not. So for temperature and sex, we see separation in the gill and muscle and liver quite nicely here. But again, neither of these tissues does like a great job differentiating between the sexes, except for muscle here at the higher temperatures. But what I want to show you here is how well it does to separate between the temperatures that bull trout generally like versus the ones they don't tolerate as well physiologically. So here in the mucus, you see these warmer temperatures separate nicely from the temperatures they prefer, the 9 and 12. Of course, the gill does a really good job. Everyone loves gill, does a good job. It is, I will say, whenever I put in a request to collect mucus, I never have any pushback on it. And I think that's something like you submit this in an AUP and it makes your vet smile. Never had pushback on this. So it does an excellent job of separating stressed versus unstressed fish in this study. When I compare these are for the target genes that amplified in the mucus. I compare this to the other 3 tissue. In our study, it's most closely related to the gill. So I should have shown you my 3D graph, but it's more closely related to the gill, and it's actually more closely related to liver, which is misleading just from these 2 PCs. But if you had it in 3D, it's closer to the liver and followed by the muscle. And you can see this more clearly here. So we see, it's closer to 90% correlation between mucus and gill, lower for the liver and then the muscle. And I'll show you here my misguided hypothesis. Liver and muscle just don't do the same thing until that onset of the cellular stress response. Then you see a bit of a similar response there in terms of like the correlation. But yeah, mucus and gill correlates really strongly, but I just heard someone tested it in the field. Tina tested it in the field and it's not correlated. So I need to do a little preliminary, but I believe it. Yeah, but it does work here. So from here, because our collaborators and myself were interested in using these chips on the wild fish, I wanted to build a random forest classifier so that I can potentially, the dream is to be able to swab a fish, measure their transcriptional response, and I can tell you, are they doing well, are they not? So this random forest classifier, you can do this in R, a lot of people do it in Python, but because it doesn't do a good job of separating between like the two temperatures it doesn't like and the two temperatures it likes, I've done an above threshold classifier and a below threshold, sorry, 2 classes. So instead of 4 classes, 2 classes. And so I run this random forest model and it can actually give me the top 10 genes that best separates between these two groups out of the 56. So what happens here is that I separate my data in, it's 2/3 and 1/3. So 2/3 is used for training here. And it has a 91% accuracy, which you can actually see here. It's showing you the misclassification. So these ones misclassified here are the 2 that are actually above but got classified as below. And this one here is a below that got classified as above. So there are always going to be some weirdo fish in there, some problematic fish that get misclassified. But honestly, 91% is pretty excellent to me in ecology. I think it's great. And then in my test, I think this test is the 1/3 of the fish. It does an even better job. So 94% accuracy on these fish. So it only misclassified 1. So some of these misclassifications, so I mentioned there are differences between the sexes in the transcriptional response. So I'll show you mucus first. Generally, we don't see a difference between the sexes within a single temperature. Where the difference lays is the shift in the transcriptional response in females. This is for mucus. It tends to occur at lower temperatures compared to the male. And for some of the genes even, we'll see like no difference across temperatures for males, but we'll see a response in the females. So there are sex-specific transcriptional responses, which I expected to see. So we see these similar trends in the gill here. So females have that earlier transcriptional shift in some of these genes. I forgot to mention these are Bayesian models. They're fit for each tissue separately in this case. So these are actually posterior means, and these are credible intervals, not confidence intervals. So if they don't overlap, they're considered significantly different. And again, we see the same thing in the liver as well. Different genes, of course, because they're different tissues, but we see in a lot of these genes, we'll see the transcriptional shift in the females is sooner. So that could maybe account for some of the misclassifications that I'm getting from my random forest models. What's interesting though is we see a reversal in the in the muscle. So in the muscle, it tends to be the males that respond sooner or have a response at all to this thermal stress. So 15 and 18, which they don't like. So moving forward, this was all done with the 56 gene version 3 STP chip. So now we've gone down to a mucus specific chip. So there are 28 genes in there that I'll be testing on wild fish. So I'm really excited about that. So these fish were collected. We started in Alberta and we have some fish in the Northwest Territories. So I'll be going back this summer to sample some more. And we actually have now, I just need to open the file and look at it, but we have a de novo assembly of the mucus transcriptome. So this will be super helpful for anyone interested in like further, this is very bull trout specific stuff, but for bull trout mucus, we can look at that. And I've chosen a subset of the males and females from this study. So I'll be able to look at just all of the genes rather than only the 56. So we can even refine this chip even further for bull trout. Although you can apply like this random forest classifier, you can apply this to any of your species that you're looking at. Super cool stuff. This is just to show you how beautiful the sampling area I'm in. But these are the little bull trout that I'm sampling. Honestly, the hope is that, you know, when you're electrofishing, mostly you're getting juveniles. So I'm hoping there are no sex effects. And with that, I just want to thank GEN-FISH for everything. Honestly, I came to Ken's lab with like no personal funding, and GEN-FISH helped me out for a few months until I could got a MyTax fellowship. So that was great. So thank you for all your support and just giving me that boost I needed to get started. Yeah, Nick.
Super cool.
Yeah.
Results. Really, really interesting. Thank you for sharing.
Mhm.
So I'm going to ask you just a kind of open-ended question here. As you know, you pick up a fish, it starts to produce mucus very quickly.
Yeah.
Do you have a sense of what's your time lag? How much time do you have to capture, you know, what's represented of the fish when it was still alive in the water?
Yeah, yeah.
As opposed to how quickly this profile of the mucus is going to change post-capture. How much time do you have to actually, you know, get the profile of the live fish as opposed to stress response from this incredibly large production of mucus?
Yeah, that's a good question. So for the lab studies, it happened right away because we're in a lab, it's so controlled, it's easy. In the field, it's a bit harder. So sometimes these fish are sitting for like 10 minutes. That's probably the most, like you were just in the field, it's a bit hard to get it like immediately. So I'm actually not sure. I haven't done a time lapse study to look at how it changes in the mucus over time with temperature stress. What I've seen though, I haven't seen a time lapse in terms, everything I focus on is transcriptional response. So I haven't seen a time lapse in mucus yet. Yeah.
Yeah, I mean, your results show that it has incredible promise.
Yeah, I'm really
Right.
excited about it. I think it would be super useful to do because we do see, like in the liver, we'll see that time lapse, like, so probably in the mucus too. Yeah.
When you did the correlation, did you do it fish by fish or did you do it gene by gene?
Oh, that one is gene by gene. Yeah, I have it fish by fish for each gene as well in like bonus. I didn't put my bonus slides here because Paige was prepping them. But I also have like heat maps of that. So generally, if we look at like the gene by gene heat map, it's stress response genes and the immune function genes that kind of light up for females at 15 degrees. Yeah.
So first of all, it's nice to see your work. And I believe Chris Wilson had his hand up. So Chris, you can go ahead and unmute yourself. Little question.
Okay. Yeah, just Analisa repeating what Nick said, that was great. Very impressive. And I think a lot of people are excited about this. I'd kind of wondered the same thing about the mucus timeline, which is why my hand came down again. But I was scribbling like mad trying to keep up. I lost track. What was the age of the fish that you were sampling?
Yeah, so these bull trout, they're actually 2 years old and they spawned the following year, which was actually quite early. So they spawned super early in a lab setting. But these ones were two.
But old enough that you've got a reasonable expectation of seeing a sex difference.
So when I opened them up, I wasn't expecting to see gonads yet because they were so young. And honestly, I opened it up and I was like, damn it. But it ended up really working out for this study. Yeah.
Very neat. Thank you.
Thank you so much.
Right.
Hello, everyone. My name's Kyle, and I will be presenting my research that I did for my undergraduate thesis here at the University of Windsor in Heath Lab. So what I looked at was comparing conventional netting with eDNA and stomach content DNA as a means to determine community composition. Next slide. Oh, perfect. So what is community composition? Well, very simple, just the diversity and abundance of fish species in an ecosystem. Now, the reason we chose to look at community composition, not to oversimplify it, but really if you want to do any sort of meaningful ecological research, I think one of the really pillars to start at is what species are in that ecosystem. And you really need to know this for any sort of biodiversity monitoring, management, or proper management, I should say, of fisheries. If you want to have conservation efforts based on actual data and make meaningful decisions, you need to actually know what's in that ecosystem. And for freshwater ecosystems, it really is an area of critical concern. Since the 1970s, I think on average, your average fish species has seen a population decline of 73%. And this is really bad considering these are ecosystems that are of large societal and economic importance. So conventionally, the way to study community composition has been through things like netting, electrofishing, and these are very practical. They're very physical, which means, you might actually be capturing the fish or shocking the pond. They float up, you can count how many, identify the species. And well, it's hard to argue that a species isn't present if you're literally seeing it. So it's very objective. But if you're trying to, say, monitor a endangered or rare species, it gets tricky because you don't want it to be invasive and you don't always want to be interacting with the species. There's another issue, obviously, with this. Well, in a pond, ponds are small, electrofishing is effective. But when you look at lakes or the Great Lakes, you don't want to go around shocking the entire lake. For one, impossible. And again, very invasive. So that is where eDNA comes in. Now, I don't really want to go too in depth explaining what eDNA is to a bunch of eDNA researchers, but it is the genetic material that is shed into the environment by an organism through either mucus, waste, shed scales, whatever it may be. But generally speaking, if a fish is in an environment, so will its genetic material most of the time, as I will explain. Now, the great thing about eDNA is obviously different species will have different genetic makeup. And when we then sequence the DNA, we can actually identify the different species and differentiate them from each other. Now, the next part of my project was stomach content DNA. Now, this is a newer application of these methods, and it works very similar, but instead of sampling the environment, we are instead just sampling the stomach of predator species. Now, this works very well for identifying the actual stomach contents with a higher accuracy than traditional methods like actually trying to identify visually what the species is. However, one area where this does come up short is identifying digested contents of the same species of the predator. So any species that displays cannibalism, this comes up a bit short because it's very hard to differentiate the DNA of a preyed upon, say yellow perch, from the DNA of the actual stomach from the predator itself. In this case, if the predator was yellow perch. So what did we actually do? Well, in southeastern Lake St. Clair, we sampled 17 locations and we conducted, or OMNRF conducted, conventional netting through gill nets, water samples connected by Dr. Matthew Yates, as well as predator stomachs. Now, eDNA, so the water samples, we followed a pretty conventional eDNA protocol here where we filtered the water extracted the DNA from these filters, and then went through the process of metabarcoding. So using universal primers to amplify all DNA present within these samples. And for my project, we went with the M-mito and PS1 primers targeting mitochondrial genes conserved through fish species common to Southern Ontario. Now for the stomach content, forgive me for the rather gross image, but it smelled a lot worse than it looks. Now, we didn't do metabarcoding for these because another issue with stomach content DNA is if you have one species with a lot more DNA present than another, it can kind of swarm the primers and not really get an accurate amplification. And if there is very little of one and a lot of another, you might actually miss the little. So we chose to do quantitative PCR using open array chips, where we targeted a 56 species assay of common Southern Ontario fish species and amplified them all independently, making it much more accurate and actually detecting those with even very little DNA present. So what do we expect to find? Well, given that eDNA has a rather high taxonomic coverage because all fish species are going to be producing eDNA, we expected it to detect a greater number of species compared to conventional and stomach content. Well, this is because conventional, through netting, has a bias towards larger and more abundant species, as those small fish or juveniles might not actually get captured by nets depending on the actual size of the net. Also, rare species in a lake as large as Lake St. Clair, which is, I believe, 1,100 kilometers squared. And even though not in this study, if you were to look at the other Great Lakes, some of them are tens of thousands kilometers squared, you might just miss a rare species. And with stomach content, we would expect it to be biased towards smaller species and have a higher likelihood of detecting smaller, conventionally prey species, where larger species aren't going to be getting preyed upon for obvious reasons. However, because the biases of conventional netting and stomach content are kind of complementary and they're opposite of each other, we expected that by combining these two data sets, we would actually create or detect a similar number of species as eDNA. So they would complement each other enough that it would actually make up for this bias in both. What did we actually find? So on the right here is a heat map, which I'll zoom into now. These are just 4 of the many species, but what this is looking at is on the left, individual species. On the top, you can see the different sampling sites, each of which have 3 methods. So the eDNA, the netting, and the stomach content DNA. Now, you'll notice just by looking at it, only one species, so the walleye, actually saw detection by all 3 methods. So we're seeing that different species are actually displaying different sensitivity to detection. So when you look at the actual number of species detected across the entire lake through all 3 sampling methods, we're seeing a total of 54 unique species detected. eDNA detected 51 of these, netting detected 26, and stomach content detected 21. So first hypothesis, there we go. That's expected. But what about combining them? This is the real interesting part. So when we look at on a per site basis, the number of species detected, we're actually seeing that there's not a statistically significant difference between the stomach content and the netting. They're actually detecting the same amount. However, when we combine them, we then see a significant difference. So even though they're detecting the same amount, we're actually seeing that they're detecting at least partially different species. So we did then included eDNA as well as the stomach content plus netting, and we actually saw the same thing. Between eDNA and the combined method, we actually do not see a significant difference in the number of species detected. But then when we combine them again, we see another jump in the number detected. So even though they're, I guess, on paper detecting the same number, so their species richness estimation might be the same, the actual species themselves are different. So this is really interesting because it kind of shows that while each sampling method has its own use, relying only on one, you're kind of missing the fuller picture. And this is really important, I think, moving forward to understand how we need to really combine these multi-method approaches for really any sort of large scale determination of community composition moving forward, to be able to actually track things like the impact of climate change, to see how on an actual site specific, or not only at the community composition level, but throughout different sites, how species move and what different areas of an environment are actually composed of. So for things like driving environmental policy and really grounding any sort of decision making in more holistic data free of sampling biases. So I'd like to thank Dr. Daniel Heath, Dr. Matthew Yates, and Dr. Michael Thorne for their contributions to this project, as well as all members of Heath Lab for answering my many, many questions and the GEN-FISH project. Thank you.
Any other question?
Yeah, I think I heard my name. Great presentation. I was just wondering, if you were to do this again, would you consider separating stomach contents by size classes and then doing meta-barcoding, or would you use the same methods as you did?
What exactly do you mean by size classes? Because for what we did, we homogenized all the stomach content, but we tried to say if there were one whole fish and then the partial remains, tried to get an equal amount of each.
So I mean, like, meta-barcode the whole fish, and then meta-barcode, like, separate all the contents into like similar sizes, and then meta-barcode those separately.
Yeah, I think that could have been helpful because for this project, we primarily used the quantitative PCR instead of meta-barcoding for this stomach content, but including something like that could add another, I guess, layer as almost another sampling method, which could show its own unique detection as well.
Okay, thank you.
It's a good idea. Oh, Nick, do you have a question?
Yes, thanks for the presentation. You know, for a lake, Lake St. Clair actually has quite strong currents. It has the St. Clair River running down, sort of driving the west side of the lake, and then it has the Thames River flume. What are your thoughts on where you sampled and whether or not the eDNA came from fishes in the vicinity of where you sampled versus were perhaps carried by some flumes? And you know, when I look at some of your species, such as Muxostoma, the red horses, I wonder if, you know, they're more riverine. I wonder if that's evidence that eDNA was carried in these currents.
Mm-hmm. I think that is definitely a possibility. And one thing I was actually talking to Dr. Yates just yesterday, or 2 days ago maybe, about was, in this case, my samples were too far apart to really do an in-depth analysis of this, but I kind of had the same question. I think further studies doing a very similar method, but keeping the samples a lot closer together. So not only to look at eDNA on a per site basis, but if you had them close enough where you could say reasonably that eDNA could be carried from one site to another without degrading in that time. You could then look at each site as individual, but also as like a group of its surrounding sites to see if there's a connection in what you're actually catching and the eDNA like around the entire area. So I think currents definitely would play a role with the eDNA, but generally speaking, at least in my study, they were too far apart to really say that could be it, because they would likely degrade in the time it would take for it to get to the next one. But I think further studies closing that gap and actually doing sites in a much closer manner could really look into that and characterize that relationship a lot better.
Okay, yeah, interesting idea. Like you could see how you could actually set up a series of sampling sites along the flume from the mouth of the Thames, or even further upstream along the south shore, and the same with the west shore with the St. Clair River. Yeah, interesting. Thank you.
Of course.
Sorry.
I was just wondering, if you noticed any like patterns in like fish were not detected by eDNA but were detected by other methods?
So there were only 3 that weren't, and there wasn't too much of an actual pattern. We're still looking into the actual species themselves. For my thesis, it was much more on like broad detection characteristics and the relationship between them. But now moving forward towards actually, again, the paper published, we're looking a lot more into stuff like that and the actual ecological significance of it, you could say. But I'll get back to you. Thank you.
For everyone here and calling in remotely, we're going to take a 30-minute break. So let's meet back here at 11:35 Eastern and whatever time that may be across time zones. All right, thanks. All right. We're going to pick back up where we left off with another 15-minute presentation. I should note that these were all this is a competition. People applied by submitting their abstracts, and all of the winners are receiving $150 cash prize. So it pays to break a leg. And next we have Jacob.
Everyone, thanks for being here. As Paige said, I'm Jacob, and I'll be presenting my research on evaluating foraging niches of Lake Huron salmonids using stomach content eDNA metabarcoding and visual identification of stomach contents. So I want to start off with just an overview of kind of a subset of the fish community in Lake Huron, just to give you a brief overview. So the species I was focusing on were 4, and there are 3. Those 3 are the leftmost. These are Pacific salmonids. On the top, we have let's see if I can get this to work, the Chinook salmon, coho salmon, and rainbow trout. These are the more nearshore salmonids. These 3 salmonids are Pacific salmonids and were introduced in the mid-1900s to rejuvenate the recreational fishery of Lake Huron. The single lake trout here in the deep benthic pelagic zone is the native salmonid, the lake trout. Now, these salmonids consume a wide variety of prey, and going through this, I'll kind of work from shallower to deeper waters. So at the top, these are yellow perch. We have alewife, which are now almost absent from the lake. Rainbow smelts, both of these are invasive species. Lake herring or cisco, these are bloater, the native species. Emerald shiner, and at the nearshore depth are round gobies and their distant cousin, the slimy sculpins, out in the benthic zone. Now, over the past 30 years, prey fish have been declining substantially in Lake Huron. These graphs show the general trends for some of those key prey fish. You can see, particularly for rainbow smelt in the top left and alewife, which have actually collapsed in Lake Huron in 2003. Their populations are quite low compared to what they were in the 1980s. So my objectives were kind of fourfold. I first wanted to determine the diets of the piscivorous salmonids in Lake Huron just as a whole. And then using those results, I wanted to infer levels of interspecific competition between the salmonids, particularly between the native species, the lake trout, and the introduced Pacific salmonids. I also wanted to compare the methods of diet analysis to combine and identify factors that significantly influence the diets of all of these salmonids. And in doing so, I wanted to specifically compare the compositions of the diets, so the percentages of the diets of each of the prey items in the diets of salmonids, as measured by visual identification and the eDNA metabarcoding. So this is part of a larger research project. Today, I wanted to focus on just these two methods of diet analysis, so visual identification and sequencing. Both of these capture the diet of the salmonid as a snapshot of up to 48 hours prior to the capture of that salmonid. So the general flow through of the lab procedures where we would get these salmonids either through angling through the lab or we would get them from angling tournaments. We would dissect the gut contents and take them back to the lab and then further dissect them under dissecting scope to visually identify the prey to the lowest possible taxon. For invertebrates, this was to order, and for vertebrates, it was to species. Each of those prey taxa were pooled based on their taxonomic group and weighed separately, and then pooled together to represent the whole gut contents of the salmonid. Those whole gut contents representing each salmonid were then put into a blender and homogenized. Then, similar to what Kyle had mentioned in his previous presentation, we did some cytochromoxidase gene amplification, and we did barcoding and sent them off here actually for sequencing at GLIR. This again reveals the diet up to 48 hours prior to the capture of that salmonid, and this particular part of the analysis is the high taxonomic resolution that we're looking for. So these are some of the prey items we found in the gut contents of the salmonids. On the top half are the invertebrates. One of the most interesting ones was this terrestrial beetle, a bark-gnawing beetle we found in the stomach content of a rainbow trout. Bottom half are our prey fish. These made up the majority of the diets, particularly rainbow smelt here and bloater were the main diet constituents. So this is what it looks like when we do the lab work. This is a stomach from a rainbow trout that was eating a bunch. You can hardly make it out in this slide, but these are a bunch of invertebrates. These are spiny water flea, which we saw in the previous slide on the top right. And let's see if these videos will work. So this is the gut contents just pooled by taxa in a vial. This is a vial full of rainbow smelts. And then we would put through the blender all of the gut contents for each salmonid and get a slurry that looks like that on the right. Then we would take our DNA extracts, PCR amplify using two universal primers, one for fish, one for invertebrates, and then we would send that off for ion torrent sequencing here at GLIR. So I'll jump into the visual identification results. These are kind of overarching the main findings we had. First, bloater was kind of the main prey item for a lot of these fish. Those are the blue bars. The red bars are the rainbow smelt. And in the left chart here, we have, based on mass, on the y-axis is mass in grams, on the x-axis for all these plots for the salmonids, we see that there were significant differences found between coho and lake trout, where coho had significantly more prey in there based on mass than did lake trout. And we also found that the mass of prey fish was significantly greater than the mass of the macroinvertebrates, which are shown in pink. Frequency of prey gives you an idea of the relative abundance of the prey in the stomach contents of that salmonid. So we see some interesting findings here. In addition to what we don't really see in the mass, we see some more rare taxa like the round goby and a catch-all bin I created of other forage fish, which I'll get into a little later. This last plot is the index of relative importance of the prey, and it's a combination of the mass, the prey, and the counts of individual prey items. So it's one metric that gives you an idea of the relative importance of the prey to each salmonid. We see this big red bar for Chinook salmon suggesting that rainbow smelt is quite important to their diets, whereas it's the opposite for coho. We see that bloater is a main constituent of their diets, and for lake trout, it's a bit mixed. Rainbow trout is dominated by macroinvertebrates, which is something we might expect. So I wanted to look at these a little more closely and try to identify the factors that influence their diet. So what I did was I converted all the data from visual identification into presence and absence data. So it would have a present if it was detected, absent if not detected. And I used simple logistic regressions to evaluate the odds likelihood of each prey item being found within the stomach contents of all of the Lake Huron salmonids. So each box shows a simple logistic regression output for an individual prey. And on the x-axis is the index. So these are just salmonids. There's 148 salmonids, and each salmonid is represented by an X on the plot. Longer sections of X's show that there were more salmonids grouped for that category of that main effect. And so each model has a couple of main effects, or some have a couple of main effects, some have one main effect. And on the y-axis, we see that there's predicted probability of the presence of that prey fish. So for rainbow smelt, we see that in the most parsimonious models, both region and fork length, so the size of the salmonid, were the main effects in the model. And we found that Georgian Bay, salmonids that were angled out of Georgian Bay had a higher predicted probability or odds likelihood that there would be rainbow smelt present in their diets. We also found that, as one might expect, the salmonids that were larger in size had an increased likelihood of them consuming rainbow smelt as well. For bloater, the largest or the main contributor to the model was the region. And for this one, it was main basin, where the salmonids were angled out of that would have had an increased predicted probability of chorogonids being present in their gut contents. Round goby, the model wasn't terribly predictive because we didn't have a lot of samples of these visually identified. So the model it had a main effect, but it wasn't significant. And the difference between the predicted probabilities is quite low, almost unusable. Looking at the macroinvertebrates, we saw the main effect in the model was actually species. So in this case, as we expect based on the previous charts, rainbow trout had the highest predicted probability of macroinvertebrates being present in their diets. So looking at the sequencing, these results are a bit different. So again, on the x-axis, we have the species of the salmonid. In the colors are the different prey groups. You can immediately see that there were a lot more groups of prey detected through sequencing. On the y-axis here, we have a scaled sequence template match count. So the number of positive hits between the library that I created of all of the prey in Lake Huron and what was identified in the sequencing. So high counts suggest there's high matches of those prey items in the gut contents of the salmonid. So we see at the top, the dark blue bars are again the chorogonids, so our bloater and lake herring. And these guys were the most detected prey group in all of the salmonids. The next largest groups were the rainbow smelt and the other forage fish category. And then particularly for rainbow trout, there's this large variance of macroinvertebrates to their diets, but one of the major constituents as well. Lower down are the yellow perch and the slimy sculpin, and one particular prey fish we didn't detect at all visually, the alewife. And then again, microinvertebrates and round goby in lower counts. So I wanted to be able to compare these results directly. So I did the same sort of models that I did with the visual identification, simple logistic regressions, except this time using presence absence from the sequencing counts. So we see immediately across all of these samples that there were a lot more main effects included in these models. That's partly driven by the fact that there's a lot more data, a lot more positive hits for particular prey in the sequencing data as opposed to the visual identification. So looking at rainbow smelt, the main effects were month, region, and species. For bloater and cisco, it was the same. We see that for round goby, it was region and month. We have more predictive models here with better distinctions between the categories of the main effects. And for macroinvertebrates, it was just month that was the main contributor to the model. This slide shows the prey that were not identified in the visual identification part, but only picked up in the sequencing. The other forage fish category includes things like shiners, white perch, trout perch, and many other fish that were just detected in such low abundance it was less than 2% that I just bin them all together as a catch-all group. We see for them region and month. Actually, all of these additionally detected prey the main effects in their models were region and month, which suggests that these two two effects are kind of the main contributors to a lot of the just variability in the diets of these salmonids. So I wanted to look and show these results again differently in terms in this format in terms of proportion. So it's more interpretable to look at these results in terms of proportion because we think of diet as like a proportion of different prey items. So this left plot is the proportion of the identified mass for each of the salmonid percentage on the y-axis. And on the right is the proportion of the sequence template match count for each of the salmonids. And you can see there are some general trends that are the same. We see that bloater's pretty big in both the visual identification and the sequencing results. However, there's a main distinction here is for Chinook salmon in particular. They had a lot of rainbow smelt identified visually, but these weren't necessarily picked up in the sequencing, at least not to the same proportion. We see yellow perch shows up in the sequencing where it was completely absent in the visual identification as well. I'm showing these results again in a different way. It's the same plot as above, but here we get a better picture of kind of the flow through of the food between the predator on the left and the prey on the, the predator on the right and the prey on the left. And this left plot is for the visual identification. This one's for the sequencing. We see a lot of it's driven by particularly bloater and rainbow smelt on the left, whereas for the sequencing, there are a lot of smaller contributions of the more rare prey taxa flowing between predator and prey. And a lot of it's actually driven by this large proportion of the sequencing reads being associated with chorogonids that were consumed by lake trout. So this is kind of very, very fresh research. This is hot off the press. I've been working on this for a very long time, so I'm excited to share these results with you. But I wanted to include the compositional data, so the percentage data in a single model for both techniques in a way that would associate the proportions of visual identification with the proportions of the results from sequencing. So this is a model that was published in January of this year, and it's called a constrained least squared simplicial regression. And we can think of that simply as just proportional or percentage regression. And in this case, I was looking at the relationship of the composition, so the proportion of sequencing stomach contents as the response, and the visually identified stomach contents as the predictors. So the predictors here in this table, and you'll have to forgive me, I'm presenting a table in slides. It's not very, very good to do that, but I haven't figured out a way to visualize these results yet in terms of the figure, because it's non-Euclidean mathematics and it's very complex. But simply here, the predictors are on this first row of data. So we have chorogonids, or categories that we saw before. And then in the first column is our response group. So the diagonals are highlighted because that's kind of the main finding here, at least for me, is that they don't have high values. That means that large proportions of visually identified chorogonids don't necessarily map up well with large proportions of sequencing from the sequencing results of chorogonids. Likewise for rainbow smelt, yellow perch, round goby, other forage fish, and macroinvertebrates. Now, this could mean two things. It could mean, firstly, that my visual identification wasn't that great, which was entirely possible. I'm not an expert at identifying prey fish. It could also mean that eDNA metabarcoding identifies large proportions of secondary diet items. And when I talk about secondary diet items, I mean the case where the prey of the salmonid is picked up, or the prey of the salmonid is picked up by the sequencing. So think of a rainbow trout eating a rainbow smelt that ate macroinvertebrates, and those macroinvertebrates are being picked up by the sequencing data, and that's why these proportions might not be matching. It could also be that we're just picking up things that are just too degraded to visually identify, and that's skewing our what we think of here in terms of correlation between the proportions of the visual identification and the proportions of the sequencing. So in conclusion, these results show there's a large percentage of overlap among the salmonids in Lake Huron. Particularly, there's this immense top-down pressure from the current high predator abundance applied to a diminishing prey base across Lake Huron. And And most interestingly, these compositional models reveal some interesting trends in the predictors of visually identified prey versus the stomach content eDNA metabarcoding. And with that, I'd like to thank a couple of people, the Neff Lab, my supervisor, Dr. Brian Neff, an undergraduate assistant, Peter Baker, the granting agencies, and the Salmon Derbies, which provided me with a lot of my samples. And thank you all for listening.
Issues with visual diet analysis and stomach content DNA is that the scDNA actually can identify back in time. What would happen if you simply went to the eDNA and removed all of the species you didn't detect visually? On the assumption that you didn't detect them visually just because they're too hard or they're too rare. What do you think your proportional correlation would do if you get rid of those extraneous ones?
Yeah, that's a good question. I would have to do it. I don't know what the result would be, but it's something I will do. Thank you for mentioning it. That's a great idea. It's going to skew things for sure, but I think a lot of the sequencing data is really driven by those chorogonids and the rainbow smelt. So I'm not sure it would change a whole lot, but it would change for sure to some degree.
If you look at your flow diagram, those weirdo ones you detected only with the sequencing, they don't look like a very big part of your analysis. So it might not make a lot of difference. And that sort of takes away from the possibility that it's just bias or in the techniques.
Yeah, and it kind of links to the other element of my research is I'm doing the isotopes as well. And the beauty of the stomach content DNA, at least to me, is that picking up these secondarily consumed prey is that it still reveals interesting information in that you're getting just a lower part of the food chain. Even though we weren't seeing the yellow perch in the visual identification, they're being picked up somewhere in the food chain of these fish. So they're important to some degree in the diet. It's just maybe further down. Yep.
I'm just thinking about salmon levels. You could also talk about potentially which one has a more varied diet.
Mm-hmm.
And I was just looking, I didn't clock it the first time, but when you just went back, it looks like your lake trout has the least variable diet and, well, at least the sequencing.
Yeah.
Maybe not with visual identification because it has a greater proportion, but anyway, my point is like, is that worthwhile noting as well?
Absolutely.
Variation.
That's a very important point because when we think about niche partitioning theory and we think about how the fitness of consumers will change between periods of high resource abundance and low resource abundance, the generalist consumers tend to do better when the resources are fewer. They can change, they're more adaptable to changes in their diet. They can find those more rare prey taxa and shift their diet to consuming those. Whereas things like lake trout, which haven't had a lot of experience competing with these introduced Pacific salmonids, they might be under threat of high levels of interspecific competition, driving them to possibly lower fitness and lower survival. Yep. Yeah, so the way I structured my bioinformatic pipeline was that I completely ignored cannibalistic consumption, which is inherently limiting because we did actually identify a couple of salmonids that were picked up within the gut contents visually, but it was such a small degree that I didn't think it was worth including in the sequencing. So when I built my library of templates that I was matching to, I just didn't include the salmonids, or the predatory salmonids, I should say. Thank you.
So I'm Chelsea Frank. I'm here to talk to you today about the development of our taxonomic and regional eDNA assays for quantitative PCR. So as Kyle mentioned, eDNA is the genetic material originating from cells that are found in the environment, variety of environments, variety of cells shed. And quantifying eDNA can allow us to monitor our species dynamics, populations, their communities, as well as map their distributions. And there's several benefits to using it, including it's very efficient where we have large quantities of species that can be identified in a single water sample, non-invasive, so we don't actually have to catch and euthanize fish to sample them, but rather minimally disrupt their environment. Very easy to standardize where we have accurate identification of species with standardized reproducible criteria across different environments and life stages. And we can also detect species that might otherwise go unnoticed with our traditional sampling methods. And one way we can analyze our eDNA is through metabarcoding or assigning a universal primer to the DNA present at high throughput sequencing and subsequent bioinformatics, or we can also use a single species approach. We do see some drawbacks to using metabarcoding, including there's misidentification in the reference database, which can lead to both false negatives and false positives, gaps in the database itself, a bias where we have amplification failure for certain primers and taxa remaining undetected, as well as we have many different protocols that are very time consuming. So in our lab, we have 4 different universal primers that we were working on designing with very lengthy protocols and multiple. And it can also produce a large quantity of data, which isn't necessarily a bad thing, but when our goal is to determine presence or absence of specific species for our end users, it could provide us with too much data if only a smaller group is of concern, say fisheries conservation or invasive concerns. Now, this is just a sample of some of our output from one of our 96 well eDNA plates. You have the species that were detected along the side, and then along the top is the different water bodies with multiple samples being taken at each. And then across this whole entire set of eDNA, we had 3.3 million reads, but only 15% of this actually represented fish species. And then with metabarcoding, we can also see a few inconsistencies where a certain species might be listed as this species or that, so we don't actually know for sure. We can use a single species approach, which is very specific and sensitive for a single species. It can still detect our difficult to find species, as well as it's a bit simpler, quicker, and more economical than metabarcoding. Now, we can use open array technologies for high throughput of quantitative real-time PCRs, and here we can actually use our single species assays to test many eDNA samples, but rather run them all together at the same time. So we can run a total of 56 different primer sets on a chip. This should be done in replicate though, so it really limits it to 28. But we can also run 48 samples due to the 4x12 arrangement. Now, there's a few benefits to using open array technologies. So assuming that we ran 4 chips in one go, it would take about 3.5 hours to prep the samples, load it, and then we'd get results pretty well instantaneously. It can also allow us to reduce the usage of our samples to just a few microliters per run. Now, in terms of GEN-FISH, we have our 3 different toolkits, the survey, health, and decision guiding toolkit. Now, the work that I've been doing specifically focuses on activity 1, where we have the development of the toolkit. So here I've actually been designing testing in terms of both sensitivity and specificity for our Canadian freshwater fishes, with some extension into activity 2, where we've been using these open arrays to test with eDNA samples from our water bodies across Canada. Now, using our single species approach, where we want to focus on a specific set of species to examine our eDNA samples, we've been using the open array technologies for both CO1 and Site B primer sets designed. So this involved looking at specific sequences, for both genes, both within and across species. So initially, we downloaded our respective sequences from NCBI, aligned them with Genius, and then targeted spaces with little to no variation to really target that specific species. And then across species, we also analyzed our alignment composed of our target gene, so either CO1 or Site B in this case, across all 200 freshwater fishes. And then there was quite a bit of trial and error to make sure we adhered to the TACMAN open array specifications. Now, this is a sample of what we would see from Genius for our Arctic grayling. We have all the different sequences for different Arctic grayling individuals, a consensus identity along the top, and then regions with high amounts of variation that we would want to avoid putting our primer sets, so we have the most cover that we can for our Arctic grayling. And then across species, very similar idea, but this time we have different species that we wanted to compare the Arctic grayling across, our consensus identity on the top, and then these regions of high variation where we actually want to make sure the primer sets are found so that we know that we're only targeting the Arctic grayling and especially avoiding those related species. Now, for deciding which taxonomic groups that we tackled, we had to compare to our existing CO1 and Site B sequences to our ever-growing GEN-FISH database. So initially, we had our salmonid and non-teleost for chips to design. And then further along, we also wanted better coverage of our 200 freshwater fishes. So we found where there were kind of gaps in the actual database and came up with the Perciform, Lucidae, and Ostereophyte sand chips. Now, when selecting these last 3 groups, we really tried to find a balance between primer sets that already existed, either from other labs or from the regional chips that I'll revisit momentarily. Now, our next step was actually to ensure that these assays work effectively for our appropriate sequences. So we tested at 4 different concentrations to determine what our threshold was for detecting species DNA. For testing specificity, we used dilute DNA of the non-target species, and this was actually done simultaneously with testing sensitivity as we ran related samples within the same chip and really focused on our relevant species. So here I'm going to show some of our salmonid results. So along the bottom, you're going to see the lowest concentration that it was actually detected at. So it was detected at that one, as well as all the prior ones before it for both Site B and CO1. And this was it being detected with really high sensitivity. So these primer sets were really good for, say, the brook trout for both CO1 and Site B. Now here we have our specificity. Along the side is the primer sets that the primer sets were actually designed for. And then along the bottom, the tissues they were tested against. We had a few that we didn't have any tissues to actually validate our primer set, such as the Inconu or the Key. And the only one that really failed when tested with the appropriate tissue was our coastal cutthroat trout. And then you can see there's some cross amplification in kind of clusters for related species for both CO1 and Site B. Now, as mentioned, we do have our regional chips. So this was our 500 water body sampling across the span of 5 years, all across Canada. And here's just a zoom in of Southern Ontario to really get a grasp of how many sites were sampled. So as part of activity 2, we had our eDNA from over our 500 water bodies, and currently we're actually working on comparing both metabarcoding results to our open arrays to determine if it's viable in terms of analyzing eDNA samples. However, we do have some limits due to the layout of the chip, so we're really limited in terms of the numbers of species we can include. But since we're limited, we've created regional chips to really help narrow down focus on a given region within Canada. But for purposes of testing with eDNA, we really tried to select species that were higher priority to GEN-FISH, possibly through invasive or species at risk concerns, and also wanted to cover species that would be more widespread, so it's more widely applicable to our eDNA samples. So for our regional chips, we have some that provide more regional cover of multiple provinces and territories, and then a few more that are more localized to specific provinces or areas, like a focus on Southern Ontario, due to higher levels of species abundance in the Great Lakes. Now, just to give an idea for our regional open arrays, this is just Labrador as an example, similar idea. Our American eel for CO1 was detected at the lowest concentration as well as those before it. And then for specificity, the majority of our primer sets worked. Some didn't work as well as we'd hoped, but we've also redesigned some since then. And then there's very little cross amplification between both CO1 and Site B for other species. Now, while our metabarcoding can help indicate our abundance of species present, it doesn't exactly guarantee you're seeing an absence of specific species if it's not detected. But in theory, using our open arrays can provide this presence-absence breakdown. And we did find some results that overlapped with the metabarcoding data in terms of no samples being found or no species being found in either. We also found some overlap between species being detected for both methods, but open arrays didn't seem to capture the full picture of the species present. And finally, our open arrays did detect some species that weren't picked up in all our metabarcoding results, specifically here the northern pike, American shad, and burbot for these two samples. Now, again, we have some limitations we might face with open arrays still, which include we did detect some unexpected cross amplifications. So these sequences might even still need to be further refined, but in cases where our species are very genetically similar, might not be as feasible. We also are limited in terms of the numbers of species we can include on our open array chips, but again, it could actually have the benefit of us focusing solely on species of interest, and we wouldn't want to include certain species that might only be found in BC on a chip for Southern Ontario, as well as we can focus on those of fisheries conservation and invasive concerns. And finally, for our eDNA sensitivity, it can be relatively low, especially since we didn't have a lot of species that were detected at our lowest concentrations. But we might be able to overcome this by running PCRs on our eDNA samples first to specifically amplify CO1 or Site B present in those samples. And I'd like to just take a moment to thank everyone that's made this work possible, our amazing GEN-FISH team, our many collaborators across Canada, and everyone here today. Thank you.
I've seen you give this talk before.
Mm-hmm.
The one thing that you didn't mention was where the qPCR assay fails. It isn't necessarily that the design of the primers and probes are a problem, right?
Yeah, so sometimes it could be misidentification in terms of like the sequences we used on NCBI or even the tissue samples we used. We only really did one tissue, one tissue per species just to kind of maximize our output for data. And if that had been degraded or low quality DNA, it wouldn't have been detected.
Some of the DNA we had wasn't great.
Yeah. Yeah.
I have a comment just because I had a conversation with DFO last week over email. That's what we had that we should talk about. But he ended up finding that tissue that were collected at the same time, different species exhibit cross amplification from qPCR, potentially because like the same equipment was used to sample and that kind of stuff. So that's something that we should also potentially.
Yeah. That'll be good for comparing like our MNRF samples to our realm ones.
Yep, we actually found the same thing in our lab. Things that were collected at the same time. And so Brooklyn and others in my lab were working out how to clean the outside of fish before taking tissue samples to avoid that.
Yeah, they were doing work on goldfish ponds where they had like that, you know, they were taking samples from ponds where there was like 10,000 goldfish, and they were getting really weird cross amplifications on qPCR. And then they actually sequenced the qPCR product, and it all turned out to be goldfish.
Yes, so Brooklyn also says cross amplification could also be caused by tissue contamination. We also have a hand up from Chris Wilson. So Chris, you can go ahead and unmute yourself.
Okay, thank you. Two very quick things. Three, I guess. One, this is really encouraging. One of the big limitations for having eDNA more widely used by management and regulatory agencies is the real shortage of places for processing. So having these chips, and especially having these customized for different regions, could make a lot of people's lives easier. So this is great to see. The only thing where I'd want to give you a bit of caution on is when you're describing the Labrador results, you mentioned presence absence. That's a phrase that's widely used in the fisheries world, and it's misused every time it comes up. So just when you're talking with people wanting to use this technology, please be very clear that you have detections and non-detections, even for quantitative relationships, because you did mention there were some species that the chips did not detect but were known to be present. That's fine. But saying absence from a non-detection is the difference between a zero and a null. And the misunderstanding is out there in the fisheries world, so you've got the opportunity to help them better understand what this technology can do. It's going to take a long, long time, but please be very clear when you're communicating these. Testing and confirming absence is entirely separate from a non-detection. So just wanted to be clear on that. Thanks.
Thank you.
So the other thing you could say, make it really clear, is the template DNA is at such a low concentration that it didn't perform a complete PCR. Then they won't have a clue what you're talking about.
Yeah, but we want them to understand.
In the original schedule, I think we had 15 minutes set aside for this, but I think I don't want to cut into lunch too much, but I still wanted to take at least a few minutes to explicitly acknowledge the immense contribution of HQP to GEN-FISH. Paige already mentioned it a bit at the beginning. I mean, 184 HQP have been part of GEN-FISH. 52 master's students, 28 PhD, I mean, more than 60 publications, and I suspect that will double before we're all done. I mean, it's just, I mean, to say that GEN-FISH wouldn't be possible without the HQP is not an overstatement. It's very true, whatever I mean to say. Yeah, and so again, just taking a few minutes to like really acknowledge that. Later this afternoon, I believe, we'll be talking a little bit about the legacy of GEN-FISH, and we talk about this with Genome Canada and the Research Oversight Committee. And we've often, when we've been talking about legacy, we talk about, you know, how our toolkits will be shared and what the uptake of our products and our toolkits will be. I think as important a legacy of GEN-FISH will be our HQP. I mean, Trevor already mentioned it, that 5 of his students and other trainees have already sort of gone out into the eDNA world, that we have a number of our GEN-FISH alumni are already doing degrees elsewhere in faculty positions, working for DFO, provincial ministries, NGOs, consulting companies. And so I would say, you know, a decade or two from now, the GEN-FISH footprint in the eDNA community, particularly in Canada, will be really quite impressive. And I look forward to, you know, 10, 20 years from now, going to conferences, meetings, talking with partners and seeing all of the GEN-FISH trainees who have really sort of taken off. And so I hope, I mean, we've seen it with the quality of the presentations that we've had today, and the hundreds or dozens, 184 other HQP out there. I hope that a lot of you will sort of keep in touch to let us know where you are and what you're doing over the next several years, because it's really going to be great to see you all come into your own and replace us old folks. Yeah. So anyways, yeah, again, can't say enough about how much the HQP have made GEN-FISH a success and a joy too. Thanks.
So Paige, I have a suggestion.
Yes.
HQP should come up with a secret handshake. And whenever you meet in a job interview or a conference, you can recognize each other.
If you give us some examples.
You do rock, paper, scissors.
Maybe I'm saying too much, but in the Lambert community, we have a special handshake.
Oh, really? My mind.
You have one and don't let go.
All right, so I have 12:21 Eastern right now. Let's still go for about an hour, a little over, and meet back here at 1:30. That'll put us about a half an hour behind our schedule, but the rest of the afternoon is mostly roundtable discussions where we look back and look at the project. So for everyone calling in remotely, an hour and 10 minutes from now, 1:30 Eastern. So see you all then. If you're here, we have lunch in the lobby. Yeah.
Thanks.
Well, welcome back, everyone. We had a nice, refreshing lunch. In this afternoon session, it's going to be a little bit more laid back. This is just our roundtable sessions where we go through each toolkit of GEN-FISH and kind of discuss. In this first day, it's more like looking back, but this is our chance to talk about the project, how it went, things we learned, things we would maybe do differently in similar projects. Just where we have this opportunity in a room with people who care about the project. And first up is activity 1 and 2. So it'll be Nick remotely and Margaret here.
Yeah, so hopefully it will indeed be a discussion, because I don't think Nick and I have an hour's worth of stuff that we're going to say ourselves. And Nick, I'll try to be aware, and certainly let me know if Nick is wanting to speak. It's often the person who's in person sometimes dominates, and I'll try not to do that. That we'll try both to facilitate the discussion. Yeah, so then activity 1, you've heard from the HQP. So activity 1 was developing and validating the fish eDNA survey toolkit, and then activity 2 is the case studies and 500 water body projects. And so we're going to be talking about, like looking back, as Paige said, sort of lessons learned. To an extent, what we would do differently. Hopefully we don't have a lot of oh, I wish I'd done everything differently type of thing, but what we can learn. So yeah, I'm not sure if you want to add anything, Nick.
Sorry, Margaret, I just popped on, so I missed the first part.
Yeah, we were just saying that it's just going to be a roundtable discussion, and hopefully that you and I don't have to talk too much, that there's lots of great thoughts from the participants as well.
Yeah, nothing to add other than I think we've learned a lot, and hopefully we'll have a discussion about that, and then have a discussion about how we can use that knowledge to our advantage tomorrow when we're talking about looking forward.
Yeah, and that's a good way of putting it. Yeah, using what we learned to our advantage as we go forward, instead of just a lot of I wish I had done things differently, and not learning from it or exploiting it. So yeah.
Grant proposal together. We said we're going to develop 3 assays for every freshwater fish in Canada. We did 2 assays for 80%. You're really in the thick of it, Mark. Do you think the 20% that we quote unquote missed, think they mattered?
Yeah, and what I will do, unless people who are online let me know that they are hearing everything perfectly, I'll just maybe recap a few of the main point. Yeah, so Daniel asked if in the original proposal we said we were going to develop 3 eDNA assays, like 2 mitochondrial and 1 nuclear for each of the fish species, and so far we've been able to develop 2 for about 80% of the fishes, and whether the 20% that we don't have done matter. First, I think that the 2 rather than the 3 isn't a big deal. It would be nice to have a nuclear assay, but for the most part, I don't think that's really critical. And I think that, yeah, we were maybe unrealistic when we said we were going to do 2 and 1. In terms of the 20% of the species that we haven't got assays for, that the tally of species was what we had at the end of the last quarter. So I think the end of March, and I think, yeah, Chelsea has made some progress on some of the remaining species. So we might be up to, close to 90% by the time we're done. Some of the missed ones are either species that genetically there aren't enough genetic differences to develop an assay, so it's not really a failing on our part. It's the species' fault that they're not sufficiently different, or a couple of species that are quite rare that we just don't have tissue or sequence for. There's a few of the remaining ones that I think are important, maybe from a conservation or a completeness point of view, that might get finished at some point in the future. But I think overall we've done quite well. I think it comes down to, what you said, that I said that what we proposed was insane. I mean, overly optimistic, overly ambitious, and I think, yeah, that we've done fairly well, if not everything we wanted to do.
Say activity 1 has done amazing. And if it hadn't been for COVID, I think that activity 1 would have nailed what we proposed, even though it wasn't the same.
Yeah, that's true too. Actually, yeah, that because of COVID, some of the initial work where we were hoping to get more of our own samples, or even maybe more from some of our partners, that where we had some identified gaps in our sequences for designing assays for some of those rare species, that partners would have been able to do some collecting. Because, yeah, it's been a while since I thought about this, but I forgot very early on, where it's just like, does the ROM have samples? Can we pay somebody at the ROM to do some subsampling? That we really had to backtrack to figure out how we were going to get those tissues for those initial stages. Yeah, and I suspect, that Nick, if we went through the list of the species that we don't have assays for, that there may be some that we recognize as being still important from a conservation point of view, and that those might be ones that are worth targeting for some like an undergraduate or a graduate student project in the future.
Yeah, I think the issue, Margaret, is you mentioned earlier, actually getting them, because you did scour far and wide for sources for those rare species, and it's particularly if they're listed under SARA and they're rare. There's quite a few hurdles you have to jump in order to even get to the point that you can sample them. So yeah, it would be useful. But it's not surprising that tissues are just not available for some of these species. And I don't know, Bob has his hand up because, back in 2007, we did a barcode of Canadian freshwater fishes, and we did not, I think we had about 185 species in that. And again, it was the issue. It's just, some of these are extremely rare fishes and very difficult to collect. I'll hand it over to Bob to follow up.
Hi, everyone. I'm sorry I can't be there in person, but appreciate the discussion. And I think one of the things that we have to acknowledge, Karen Helbing's team on the iTrack DNA project just had a paper come out today with 125 different assays. Spanning, not just fishes, but other taxa of concern. And I was a little surprised in that, they say we followed the CSA guidelines. They've used whole mitogenomes to search for optimal gene regions that can tell closely related species apart. And then there's a little passing note about, and, we don't have great coverage for a lot of these things. I mean, what we tried to do in GEN-FISH that was a huge undertaking was to also account for regional variation and not just sample an individual. So I do think that was the right way to go and made it even more over the top ambitious. But I mean, just even trying to circumscribe what species occur where and whether or not there were tissues available for them. I mean, that really is the right way to go about doing this, rather than, in my opinion, and I'm not trying to speak ill of anyone else. We can only do what we can do with the data we have, but kind of going around saying, well, we've got a whole mitogenome from one individual of all of these species, and we're going to make an assay that seems to tell them apart without taking into account, intraspecific variation regionally is potentially dangerous. So I think, what we tried to undertake was, maybe a little unrealistic and hampered by COVID, but what's been accomplished, even just in summarizing what's where and what sequences we have, was pretty significant in my opinion.
Yeah, that's a really good point. And I think we saw this in Andrew's talk as well, like with the bull trout versus the lake trout, that in some parts of the range, the assays are species specific, but not all. And we'll certainly see this tomorrow when we go over the activity one sort of high points, that a lot of the published assays aren't universally species specific. They're only going to be species specific in particular regions, or a lot of the published assays are maybe the easier ones, the species that are much more challenging because there is a lot more intraspecific variability and a lot less interspecific. And so that when you're trying to do it across the species range, it gets much more difficult. And some of the really closely related species, it takes a lot of work to get a species specific assay. So I know I was mentioning in the car yesterday coming that some of my first foray into eDNA was with sea lamprey, which is the only lamprey it's the only lamprey in that genus, and lampreys, as we know, are very distantly related from everything else. That was dead easy to make a species-specific assay. First design, it worked, and then we spent 3 years trying to get a species-specific assay for carmine shiner. That it was just a lot more challenging because there's a lot more closely related species in the region, and so we've been very ambitious, but that's because we didn't want to just cherry pick the easy ones or particular regions. We really did want to try to provide resources that would be broadly useful, and then coming up with entirely species-specific assays for all of the species, being very transparent about what it cross-reacts with, and so that if you are assaying for bull trout in an area that lake trout doesn't occur in, then you're good to use this assay. But if there is the possibility of both, then you can't use it, et cetera. So being really transparent about what the potential drawbacks might be. Yes, and I'm glad Brooklyn brought that up because speckled dace falls into the same category as carmine shiner. It's not one of the easy ones, and it's one that Brooklyn has devoted a lot of effort to so far, and I'm optimistic it's going to pay off. But it certainly wasn't just a quick let's work through the list and we can check that off in a couple of days. Yeah, it takes a lot of effort to get something that is species specific.
Margaret, I was going to weigh in on that as well. And this sort of bleeds into tomorrow, because where we're having some of these challenges, and speckled dace is known to be a species complex, at least in the US, that has not yet been described. Where we see some of these challenges are I would consider to be potential areas for further taxonomic exploration as well, to confirm that the challenges are not actually at the species level, that in fact there are more than one species.
Yes, that's a very good point. And then it also makes me think too, like a lot, if we just go with sequences on GenBank, if we're getting like speckled dace sequence from the US, it may be very different than what we would get in the Canadian portion of the range. And yes, and I know lampreys better than I know speckled dace, but in some with all of the lamprey paired species, we couldn't design truly species-specific assays because there's taxonomic uncertainty about whether they are indeed different species.
So I propose that activity 1 be used for taxonomic reclassification of Canadian print plants. We can't produce an assay for them. It's not a species.
I think the systematists might be upset with that declaration. You know, the thing is, like, where we had some issues was, you know, when we were actually coming up with species identifications, often there would be American species in there, and we end up having a filter to say, here's the Canadian species list. And of course, that's great if you're expecting native species, but if you're trying to find a previously unknown invasive species, that doesn't help. But you know, you take a species like Persina caproides, the log perch that, you know, 30 years ago was one species throughout its range. I can almost guarantee if you compare a log perch sequence from Canada against GenBank, it's going to be some log perch from the southeastern United States because they're very similar. And I was looking at some results yesterday that Matthew had sent me, and there was one there. But you know, genetically, they're so similar across these, probably a 10 species complex now. And, you know, so that's, you know, one of the challenges of confirming your identifications is if you use something like GenBank or another online library, the majority of the specimens are not from Canada, and many of them are misidentified as well. So, you know, this is part of this challenge of, and we need to figure out what, you know, Notropis lipomus as an example. We need to, you know, we need to figure out the reasons why we are not able to identify some of these species to what we think are species. And there's at least 3 explanations that I've just brought up.
Yeah, and I think this is one of the great strengths of GEN-FISH. We have such a range of expertise on board, and so that, you know, not in addition to sort of the technical genetic expertise, having the fish expertise, and so that it's not just a matter of downloading any sequence from GenBank and assuming 100% that it's accurately identified and not paying attention to the location of collection, really making sure that it all sort of makes sense and being sufficiently distrustful of online data to make sure that you're not ending up going in the wrong direction. For additional comments and questions on it?
BC speckled days is more similar to BC leopard days than Californian speckled days genetically. And then Markel jumped in to say Notropis and Bahamas in GenBank are messy.
Yes, yeah. So some of the shiners and sunfishes, messy as well. Yeah, and so some of it, I think, potentially misidentifications, but also, as Nick was saying, with species complexes that may be confounding things.
Well, part of the problem is NCBI, GenBank. Not many people here will remember, but there was a time when people put on their CV that they had uploaded sequences to GenBank because it was considered an accomplishment. And when you start getting this intersection of technical incompetence and taxonomic incompetence, of which I'm a prime example, you really start getting problems with faulty sequence as well as faulty fish identification. So, you know, Bob, your lab was doing phylogenetic trees to look for outliers for faulty sequences. That's the way to go, but man, that's a lot of work.
Yeah. Doing things properly takes a lot of work, and that's not always the case. So being sufficiently skeptical of all data to make sure that we're not being misled.
Except ours.
except ours, but because we're going through all of the checks, we're being skeptical of, not skeptical, but critical in a good way of our own data, that we have rigorous checks and balances.
So in what way?
You need blanks now. Early on, you can publish DNA with no blank samples.
Yeah.
So that's one that really jumps out.
Yeah, because I would say, for the question about, whether, publishing eDNA work, that it's more rigorous. I think a lot of it will still depend on where you're publishing it, who is the handling editor, who is the reviewer. Matthew?
Yeah. I had a lot of discussions about it actually last year, and it's something that works to basically have a series of standards that basically papers have to meet for even to be considered. And of course, the blanks and negatives are one of them. There was a paper that was submitted this year, actually, that stirred a huge amount of controversy because it was this massive effort to do an eDNA survey and a C, and it was a great study, but it had no blanks. And it caused a lot of problems in the review process for it. And I think that it's probably going to get through this time, but I think it's going to be the last paper that manages to squeak through those types of standards in place.
Yeah, interesting. Yeah, so I'll just recap that in case everybody online didn't hear. And so, Matthew was saying that he's on the editorial board for environmental biology of fishes. No, sorry. That's environmental DNA. Sorry, have it. Those three words just come rolling out. Environmental DNA. And there's been a discussion, about whether there is essentially a checklist of features that every study has to have to even be submitted. And a recent paper that was, quite a large scale survey, but that didn't have negative controls. And so, what to do. And that's, I think, especially for a journal like Environmental DNA that is purely focused on environmental DNA, you need to have those standards. I suspect that it will be a lot looser, hit and miss in other journals, like a fish journal that might occasionally publish eDNA papers. But having that checklist of it's not good eDNA research if it doesn't have these standards.
Speaking of big assay or project, how about that 500 water body project? Okay, that's got to be the biggest one that I know of. Have you seen anything bigger?
There was one that came out in England that was not 500 water bodies. I think it was closer to 100. That was comparable in scope. 500 water bodies is still bigger.
Yeah, but you could put England in Ontario.
Exactly. Yeah, sure. Yeah, it's like the highest.
And remember, their water bodies are ponds.
Yeah.
Yeah, that was it was amazing that we were able to actually get 580 in the end. I'm stealing some of Matthew's thunder from tomorrow, but it was just again, like activity one, it was a slow start because of COVID. We were delayed a couple of years. We did get out and do some solo, what was the word again? Geez, it's so long ago, I can't remember when you had to bubbled cruise. We had bubbled cruise, right? But we only had so many of them, and a lot of the partners who we had hoped would go out could not sample at all. And so we had a slow start, but I would say a roaring mid. And by in the last couple of years, we were just then cherry picking, trying to figure out areas of Canada that had not been represented. So, you know, I you know, looking back, some of the challenges were just getting the samples, getting them back to the lab, getting people to sample in a standardized way. Matthew can speak to getting a master database of all the data that went with those samples. You know, we're at the end, but that's all of that came together for the most part. I don't think we, with activity 2, we didn't do everything we wanted to do. For example, analyzing the output from the 500 water body survey, but I anticipate that we'll see that happen in the next couple of years.
Yeah, the output for that will be vital for quite a few questions. And I mean, I'll talk about that some more during the presentation. I've got some ideas on what we can do with it that I think are probably unprecedented and not really able to have been done before, just based off of the sheer scale. And one thing I'm really interested in, you had to talk about this earlier in terms of assay design, but also in terms of species identification, is just haplotype variation at a biogeographic scale. And so that's something that your postdoc Alex actually is expressed interest in working on, just to be able to use the metabarcoding data set to discover, new haplotypes and intraspecific variation across Canada. Yeah, for all the populations that have it.
So Matthew, those are great ideas, but since we're supposed to be looking back right now, I do have a question for you about the database. What could we have done differently so that it was smoother?
Yeah, I think probably, so one thing that we did was rely very heavily on kind of community organizations to do and grad students to do a lot of the sampling. And I think that we could have done better on being a little more forceful is not the right word, but yeah, draconian also isn't the right word. Yeah, but basically
authoritarian,
being a little more stringent in terms of how we
Brooklyn's like unified.
Yeah, unified in terms of how we try to collect that data, but also to try to do it in a little bit more of a top-down organized manner. I think that's the main thing. Organization of all of that turned out to be one of the biggest challenges that we face, and kind of coordinating. We tried to get it coordinated so that people reported data in a sort of standardized manner, but it was something that was difficult to do. I've got some ideas on how we might do that in the future. I don't know how many more 5.5 water body surveys that we participating in the future, but yeah, definitely a lot of things to that. But I think that's a big one. It's kind of just trying to be a little more organized and
perhaps also if we had the resources for a dedicated database manager along the way.
Yeah.
The lack of standardization would have been caught earlier and corrected. I'd probably, and there's also ways to force standardization through, various, forms and so on. But, the trade-off is you may turn potential partners off of doing it if they feel that the work is overly onerous.
Well, yeah, the thing that's very interesting about it is that, yeah, I think that's actually a very, very valid point in terms of like getting buy-in, because what we did was only possible with buy-in from community organizations and a lot of academic labs across the country just volunteering samples. And so, like, we got samples from a lot of provincial agencies, and the samples from those agencies almost invariably were very easy to work with, because they have very stringent metadata. They have protocols in place already for how they can handle those data. And but they're challenging to follow. Like they take a lot of work to follow. So if we're just asking, you know, like the Saskatchewan Association of Community Watersheds, or, you know, the Swan Lake Conservation Group, like all kinds of different groups participated in the survey to really kind of follow provincial and federal level data management practices is a pretty big ask. I mean, truthfully, so there does have to be some level of kind of assistance with that. And I think, yeah, what you mentioned before, having a sort of structured, maybe structured forms might have worked out well in those situations. But yeah, it is. I think we could have run the risk of scaring them a lot more years by being too emphatic on some of the things.
Brooklyn says, from an assay design point, if she could go back in time, she would have suggested to do a deep dive into assay design, develop a standardized method, and then have grad students follow that method for consistency.
Yeah, that's great. That's it's like Brooklyn and I on the same page on so many things, because I was going to say, yeah, like sort of building on what you were saying about having things more standardized at the beginning for activity 2. I'd say that in hindsight, I wish we had done that more for activity 1, where we had more of a somewhat standardized sort of workflow for assay design and validation. At the same time, sometimes you have to try a number of different things to find out what one works best. And so we didn't know what one works best in year 1. Maybe we only got there in year 4. Maybe year 2 would have been better. But yeah, that I think there was a lot of learning that went on, but also maybe some lost time as we were sort of finding our way with the assay design and validation. Yeah.
They actually detail their workflow for designing assays. And I read that and I was like, man, this would be like, that is like a grad student starting and, you know, just having somebody like read a very standardized protocol. It doesn't have to be like the standardized protocol. Things are always going to be different, but just having a document of different like steps in very great detail. So I think that might be like a genuinely valuable potential input.
Yeah, and the validation paper that the HQP started on, that's almost ready to go out the door, it sort of is a bit of that, where it's not as detailed and prescriptive as some that are out there, but it's meant to be a lot more user friendly, so that some guidance, but not your assay's crap if you don't do exactly this, but a lot more sort of structure and guidance. Yeah, with 3 case studies, optimizing and validating a published assay, you know, making it work for you, a genus specific assay and a species specific assay, and sort of working through those 3 case studies. So any day now, that'll be ready to submit to environmental biology of fishes. It's not environmental DNA, but yeah, but I think that's what's needed to break down the divide between the eDNA experts and the people who want to use it, and that it's sort of too hard for the people who want to use the eDNA assays. If there's not a perfect one or a good enough one already published, they don't know what to do. And so allowing some more of that optimization and validation.
So for large scale studies, such as the 500 water body, one of the things that's possible is citizen science. We did that when we were doing bacterial eDNA, but it was just in southwestern Ontario, and all they did is gave us their Latin log. And then we were able to mine water quality data from online. So this question is actually, Sapna, if you're there, do you think we could just sort of, instead of getting the sampler to try to come up with water quality, we could just get Latin log and then use existing databases to find out what's going on in the water body they sample?
Yeah, that's a good question. Are you talking about this in terms of fish or in terms of water quality and climate and morphology indicators?
I guess just metadata in general, because if you did like a national level eDNA survey for fish, and you did it as a citizen science where you mailed out kits and people send it back to you, they're not going to even get reliable temperature data, much less secchi depth or fish community. So would there be any value in just getting these filters mailed back if all you had was lat and long? Could you fill in the metadata?
Yeah. For some lakes, you can. If they're greater than 0.1 square kilometers, you definitely can get access to all of that data. You can get information on all of the morphological characteristics, water temperature, ice, mixing status, etc. Maybe nutrients, but they have to be larger lakes. I think that's the caveat that we've run into. So small lakes are really hard to do. And we can benefit from satellite records to get metadata. So anything greater than 0.1 square kilometers is pretty good for water chemistry as well. But I like value the power of citizen science, community science data. I think it can be really helpful in especially evaluating areas that are harder to get to, but also to look at some of that spatial heterogeneity within a region, which I think can be difficult to do over a broad scale. So I think it's a nice idea.
Chris
That's your question.
Sorry. Chris says what Daniel's describing sounds very much like the US eDNA Atlas.
That's sort of the US eDNA Atlas is what I was thinking. I don't know what they did to overcome the issues of lack of metadata.
That's a good point. Yeah, because I'm only familiar with it, I think, with some of the quality control checking that goes on before they share data collected by either citizen science groups or people sort of outside their own sphere in terms of I guess it's kind of like we were talking about with GenBank. You can't, if various people are submitting data, there's no way to be 100% sure that what they're submitting is accurate, but there's at least sort of a checklist. You know, did you do this? Did you do that? But I don't remember anything about the metadata apart from like latitude and longitude.
Also, I think it depends on the reason why you would want to do this as well, right? Is it simply to increase our knowledge of the potential distribution of species, or is it to further evaluate the utility of environmental DNA as a potential monitoring tool or tool in our toolkit? Where for the former, maybe you don't need anything more than a lat long, but for the latter, you may want that additional information. And again, I think we had envisioned that as a part of GEN-FISH that we would have actually done the analyses to determine what additional data would be important because it may influence the results. Again, we didn't get there by now, but I think, like as Matthew was alluding to, this is not the end of the analysis of the GEN-FISH data. And there's a lot of questions there still. I'm sure that some of us will continue doing that in the future, and I look forward to having a fuller discussion about what the future holds tomorrow. But again, I think we had in mind that we would use this data set to determine what data you absolutely needed in addition to say lat long, to report back on the findings from an eDNA sample.
Since we started GEN-FISH, we were responsible for putting together a report page and Joe, and then we would present our progress to the Research Oversight Committee. And activities 1 and 2 always got focused with a lot of attention. And it was almost like you were doing a defense all over again, a couple of times a year. Can you share with us some of the critiques that you commonly heard, positive critiques, let's just say, in terms of what the outside community, academic community felt about the project? And typically some of them were international, some were local, and so we kind of had a diversity of opinions. If you can recall, but if you blanked it out on purpose, I also understand that. But if you could have any recollections and just to show everyone in the room what we have to contend with in terms of always justifying our existence, so that we can get GEN-FISH to continue on.
Yeah, that's a good question because I had largely blocked it out. But yeah, thinking back, the biggest challenges initially it was twofold. I'll talk about activity 1, then Nick can talk about activity 2. With activity 1, we were met with considerable disbelief when, and I'll talk about this tomorrow, Stephen Rogers at the University of Guelph, working in Bob's lab, had done in silico testing of published CO1 assays and found that only 5% of 101 published CO1 assays were truly species specific. Another 20% were regionally specific, but the majority of them were not. And it was kind of what we were talking about with, the lake trout, bull trout, that in some areas it might work, but it was only because a lot of the published assays were tested against a relatively small subset of other of non-targets, and so that there was some skepticism that the published assays weren't as good as advertised, and then some maybe surprise that it was taking us so long to fill in the list of the remaining 95% of 218 species. But as we're saying, it takes a lot of effort to get a validated species specific assay. It's not, the first primer and probe sequences that you design aren't necessarily the good ones, and all of the sequencing that was required to even design an assay, and all of the reference tissue collection to be able to properly validate them, that a lot of that initial effort took a lot of time before we were starting to really see our assay tally increase. And so I think there was some skepticism that the published ones weren't as good as advertised, and then some, what is wrong with you people that it's taking you so long to design the rest of them. And then with the open array chip, and a lot of efforts over the last year, we really did pick up the pace. But and again, I mean, it was far more work than we ourselves anticipated it to be. And Nick, you can talk about
Sure.
activity 2.
Sure. Well, I think some of the initial impressions from the ROC were that 500 water bodies was very ambitious, but I think that was deliberate on our part, right? We wanted it to have a wow factor and stand out, and seem like it was a number that may be challenging to get, right? And early on, again, because of delays in sampling, there were concerns of whether or not we would get 500 over the course of the funding. And then that transition, when we started filling out the maps with points, and you'll see the map with points by year tomorrow. Then it went from, will you get to 500? Is now you need more complete spatial coverage, right? And then, it was a little more challenging to find opportunities to get samples, for example, from the far north, right? The Arctic and the subarctic. But they pushed us to do that, and we found collaborators to help us with that for the most part. And then on the case study side, I don't recall many comments about that, but the one comment that came up over and over again related to case studies was if where we had Indigenous partners, how did we engage with those partners in order to co-produce the case studies? And we thought over several meetings that we had done a good job of explaining how, but we kept on getting the same comment that the explanation was not sufficient, despite the fact that, so there was a mismatch between how we thought we had done in terms of explaining how this knowledge co-production occurred with how the ROC perceived it as occurring. Would you believe it took till the very final meeting for us to actually nail the explanation? Paige came up with this master spreadsheet that allowed us to explain for every one of the projects how we undertook knowledge co-production. And I think that the ROC was finally satisfied with our explanation. Had we only thought about that 3 meetings ago, that I think our life would have been a little easier. But you know, I thought we had done a very good job with knowledge co-production. We did not do a very good job, obviously, of conveying that how we actually did it. But this last table at the last ROC worked, and it appeared I don't know if we received the ROC comments yet, but it appeared that they were satisfied with the table. In fact, they explicitly thanked us for including that table.
And I think, yeah, the ability to get so many of water bodies speaks a lot to our collaborators. And so again, one of the great strengths of GEN-FISH is that it wasn't, I mean, it was quite a network of PIs within GEN-FISH, but then also when you factor in all of the collaborators of the PIs, it really it was a very powerful group.
We should also remember that in 2017, 2018, when we first started thinking about this, eDNA was much more in its infancy, right? And there was a lot more uncertainty than there is now, and then trying to get collaborators, partners, particularly, say, for example, government agencies who have to make at times legal decisions based on data, to buy into, at the very least, participating in this proof of concept project. I think it was a little more challenging then than it would be now. Interestingly, I work in South Africa as well, and they have a 3-year eDNA project that is like a mini version of GEN-FISH, a very small version, but they're still at the stage where they have to convince the managers that eDNA is a valid sampling method. And, they have to do that by doing the side-by-side sampling in order to compare the results in order to convince the managers. So we've come a long way in Canada, but I think we should remember that at the beginning, there was a lot more uncertainty about eDNA than there is now. And hopefully, in part, there's less uncertainty because of GEN-FISH itself.
And more substitutes.
One thing that I've heard was a real interest in the custom open array eDNA chips. And that was sort of thrown in, was not originally part of GEN-FISH. And, that addition, sort of like the non-invasive sampling we'll talk about in activity 3 and 4. It just, again, it never occurred to us when we were developing the program, the project, even though we had the open array in activity 3 and 4, But it certainly has generated a lot of interest.
Yes, I'm glad you mentioned that because if we fell short a little bit in not delivering 3 assays for all 218 species, yet we've certainly added a lot, more water bodies. And yeah, you're right. Yeah, the open array chip, because yeah, it's that sort of, like, Goldilocks, like the single species assays not quite enough if there's a number of species that agencies, et cetera, are interested in surveying for. And then metabarcoding is more than they want, and that the open array chip being able to simultaneously survey for a custom suite of 28 or 56 species. Yeah, I think there's going to be a lot of interest and uptake in that, particularly if we've really well validated those assays that are on the chip. That I think, yeah, it's going to be a really important contribution.
And to Tina's comment about the Research Oversight Committee, one of their comments is we're out of our minds not to commercialize these and get some benefit from selling them.
That's a very expensive shirt you're wearing.
Well, Dan, time to trade in that lab coat for a suit.
The time to move on?
It can be. That's gonna be going.
So, did he leave? There it is. Yeah, that's it.
All right.
Back and forth. So I'd like to say that in my opinion, the greatest accomplishment of activities 3 and 4 is hearing Nick Bernier say that he thinks it actually might have some validity. Because since day one, he's been saying transcriptional profiling with open array chips isn't going to work. And now I think you're a convert, or at least partially. Seriously. So do you want to say something? Let's start with activity 3.
Okay. Yeah, it's a lot to reflect on over the past several years. It's been, it was a lot harder than it sounded to produce all of those chips. Learned a ton, which is good. Pretty excited about how they've been now applied to some of the studies as part of activity 4. So activity 3 was really nuts and bolts and technical, where activity 4 is where the technology and the approaches really got to shine. So pretty excited about all the different ways that they've been used over the last several years. And we're only now starting to really take off with all of these publications coming out. So it's really, this will have a nice legacy for the next several years for sure. And to echo what was just said, I mean, things could have been done more strategically. COVID did not help when you're trying to be in the lab testing things. And one of the funnier hurdles that COVID gave us was that at least the province of Manitoba was using the same technology for doing all the COVID screening. So we couldn't even get the materials we needed to then do the work in the labs, even once we were allowed to return to the lab. So there's a lot of interesting and stressful moments along the way. But yeah, just really impressed that it's worked, and really excited by all the different ways that we're using these things now.
Yeah, we did we meaning Ken and the group that developed the assays did a lot of pivoting, because as was mentioned earlier, the original plan was one universal chip for all fish species. On that one, Nick, you are right to be skipping. So we had to change directions pretty quickly on that. But the other challenge I think that activity 3 and 4 had was the concept of environmental DNA was very appealing and obvious, maybe not obvious, but acceptable to managers or potential end users. It made sense. Okay, the fish are dumping DNA and you're using magic to find the DNA. That is very sort of straightforward. But as Tina can comment, some of our partners thought that the chip was something you put in the fish and it radioed back how healthy they were. And so that the disconnect between what we were doing in the lab and what our potential end users thought it could be was a challenge. But on the other hand, we've got people who are adopting this, and Chris Wilson is one of the earliest of the government embedded people. But Climate Change Canada has contacted Ken and I about applying these chips for projects in the Great Lakes and the Arctic.
Yep. Yes. And so, yeah, it's generated a lot of interest especially from government labs. Analisa mostly has been working with folks out of BC as well, doing some salmon work out there using the chips. And then we've typically get a lot of attention at conferences too. A lot of people come up afterwards and ask if there's ways that they can work with us and try to incorporate that approach into their analyses as well. So it's generated a lot of attention. More so on my end, it's been more so from government and academics, less so from consultants or anything like that. But I think you may have heard more from that side of things.
A number of consultants are very interested in following the technology because they would love to be able to go in and capture some non-critical species in areas of environmental concern, have Shelby run the STP chip, and then report the fish are not showing any signs of contaminant stress or thermal stress or whatever, sort of before and after development. Of course, in Ontario, they don't have to worry about it anymore with Bill 5. Yep.
How do you think the GEN-FISH chips fit in with people who are used to using something like DFO's fit chips?
So yeah, the question is, how does the GEN-FISH STP chips relate to DFO's fit chips?
So in full disclosure, I was co-supervised by Christy Miller for my PhD, who's produced the fit chips. So we can have very candid conversations about our different approaches. So the fit chips are just basically qPCR assays that we're using a fluid ion approach, which means they can produce they put out the probes and the primer combinations themselves. So it's a little bit more flexible in terms of like what they can run on each individual fluid ion chip. We can run more assays at a time, which is a nice plus for our side. But their work has been mostly on Pacific salmon. And they started out doing multi-species chips, and they've now sort of switched to species-specific chips, and that are more targeted for specific types of stressors that are relevant to Pacific salmon. So it's very similar approaches, but we cover a lot more species that might be of more interest to the rest of the country and less Pacific salmon specific.
Most common chip species?
Well, the salmonid chips seem to be really popular. Yeah, so I've pitched this idea to Christy Miller's group about let's take the same samples from the same experiments and compare our approaches just to see, not as a comparison or anything, but just to see like how well both of them perform for trying to identify these types of markers. Again, just it's better to work together than it is to try to compete against each other.
this. To me, the big surprise with the STP chip is there's been very little interest in it in fish culture. And I really thought like the Normandale hatchery here in Ontario would simply go for it because it would be an objective, fast, relatively inexpensive way to monitor health of captive fish populations. And if you're growing 1 million smolts, you don't really care if you have to kill a few. But the BC Freshwater Fisheries Society are really interested in using environmental RNA. And I don't understand because they've got thousands of fish in each tank. It'd be so easy to pick one out and sample gill or sample liver, but they really like the idea of taking a water sample and getting an average transcriptional profile for all the fish in the tank. But other than that, we haven't had a lot of interest in using it for fish health in large scale culture. Anything else?
I don't know. I covered most of the things.
Yep.
I have to reflect on. So yeah, I think just the big thing is that, you know, these assays have now been produced and, you know, maybe they're not perfect. They'll continue to be refined over time, but now that we have them ready to go, we'll be able to more easily incorporate these chips into future studies for sure.
Is there online?
Chris, do care a lot for stocks, so you may still be interested in fish health chips in the near future.
Yes, sorry, Chris, I wasn't indicating that they don't care about health. It's just that they're, I would say, skeptical about this technology. So they prefer to stick to behavioral and necropsy approaches to assessing fish health. That's my impression, although it's not based on anything they've said to me.
Daniel, do you think it could be just a side effect of not all of our PIs are working with fish culturists, and that could be a reason? I know we have got the BC Freshwater Fisheries Society, we've got some, but across Canada, do we have that? Like, is it our fault that we're not bringing it to the fish culturists? And so instead of putting it on them, because you just said like some of our partners thought that the chip was like a microchip.
Mm-hmm.
So potentially their reticence isn't a reticence, it's just not knowing about it.
Yeah.
Or am I on too much?
No, I think you're probably right. Although the Ontario system does know about it because Trevor's been working with Normandale to use it to assess stress in the transported Atlantic salmon.
Very good.
But it might be just a delayed uptake.
Yeah.
I think that might be happening a bit in Manitoba as well, because we've sort of had some changeover as to who's running the local hatchery for salmonids in this case, and the new leadership is kind of more eager to incorporate research into what they're doing, and so they've already reached out about trying to bring some of these approaches for monitoring their fish. So hopefully that'll pan out in the next few years.
Margaret, you had a question earlier?
Fewer people can run the open array chips in their labs, and the infrastructure for doing so is more expensive than just a typical qPCR machine. So if this takes off, both open array for eDNA and for fish health, is this something that you would envision, say, consulting companies being interested and able to offer, or is it something that could be done on a cost recovery basis within academia?
Well, I mean, it's set up really nicely here at Gleer in terms of running assays that way. I've pitched that idea at U of M and met with some resistance over get into it. I met with some resistance, but I think it would be great to do it in a cost recovery approach. But I mean, at least for the fish health toolkit, we are making the assays publicly available, and so people who don't have the machine can still benefit from what we've done as well in the future.
Shelby.
It's just a matter of people knowing that that's available.
Yeah, so in case you can't hear online, Shelby's the manager of the EGF here at Glear. And she was saying that she is actually doing fee for service with some consulting companies at this point. And certainly some of the project consultant company partners have indicated interest. And one of their biggest concerns is access to having the assays done. So having the actual open array machine available for getting the samples run, because not many consulting companies want to get into extracting RNA and making cDNA. Extracting eDNA doesn't seem to bother them too much, because you could sort of do it in a small eDNA in somewhere in the lab, but the RNA seems to be more daunting. Nick, you had your hand?
Yeah, this is just more of a general comment, but I think we've come a long ways from coming up with a list of our favorite genes. You know, this PIs on the grant and 112 of our favorite genes to realizing which ones could work on this platform, but which none of us had to work with. We didn't know the limitations of the open array. We had knowledge about qPCR. It's a learning curve just to get to understanding what the limitations of its technology, the limitations of our favorite genes versus the genes that can work, our limitations of within different orders of fish, what we could get away with couldn't. As it turns out, we can do all sturgeons on one chip and at a very, very high efficiency. Over 95% of the genes work across sturgeon species, and likely that would be the case for some monids. As we move to larger orders, I mean, we knew it was somewhat utopic, but still we had to come to the realization and it takes years to do that.
Yeah.
And similarly, I think with the adoption, for example, with like fish culture, if you can provide evidence that you can detect a dysregulation at the transcriptional level before an outbreak of disease, I bet you that people will be lining up for the chips. But we have to get to that point where we can show this profile means that your fish, give them another week or two and you can have an outbreak. Okay?
Yep.
But we're not quite there yet, but we're getting there.
So a couple of things. So hopefully people online heard you. I could hear you clearly. But first of all, your comment about our favorite genes. So we were writing the proposal and we had to be specific. So I sent out an email to all the people I know who did qPCR on gene transcription and said, what are your favorite genes? That was actually the wording of my email. And that's how we collected. But we had to have a target set of genes that was based on something that had a background. So, you know, Dixon had been working, actually Dixon and I, on a series of immune function genes. And we just knew they actually worked at some level. That if you really kicked the fish immunologically, those genes would change. They would show a significant response. We didn't want to include genes that were basically, you know, completely insensitive to the environment. And so that was the favorite gene part. The original proposal was that we were going to pivot from that. We were going to test the 112 and say, okay, 80 of them worked,
we got to come up with our next favorite genes for the 32 that didn't. And we just got derailed, right? We couldn't test anything for 2 years, more than 2 years.
I just add to that, you also want to be sure that you're targeting specific stresses as well. When you're mentioning disease of like low hanging fruit, we were talking about, we looked at the WWF Wildlife Fund of Canada and the issues facing freshwater fishes. And obviously it was disease, ecotox, it was aquatic invasive species. And it was like, yeah, we're going to come up with a health check that can identify if they're battling AIS. Then it was obviously temperature and competition. So that was also a challenge to find favorite genes that can also be specific enough to reveal the stressor that they're facing in a prognostic way, so not necessarily a diagnostic way. And that was another hurdle. And so I don't know. Oh yeah, and then obviously osmoregulation. I'm just forgetting some of the functions. That was a challenge too, when you say in terms of trying to nail it to identify a specific stressor, when you've got a general stress gene, and then you've got an anoxic stress gene, or you've got an osmoregulatory stress. AIS stress gene might still be on the fence, but I was wondering, like, so yeah, there's much more mixed with that in terms of what we still need to potentially prove, or, you know, what are your thoughts on that?
Well, yeah, I mean, that's always been a struggle trying to sort of separate out the general stress response versus something that would be specific to a particular stressor, especially, you know, in the grand scheme of things, that would be relatively few numbers of targets for these types of stress responses. But I think a lot of that will come from all of the studies that are going to come out of Activity 4, which are going to produce these gene transcription profiles that we can then really start to mine for trying to determine what is a characteristic response to a particular stressor in a group of species. But that's, you know, it's down the road, unfortunately. We had plans on doing that for this one, but, you know, I think once we produce enough data from all the studies that we have going on now, we'll be able to start really diving into those types of questions.
Yeah. Oh, yeah. So the ROC, they basically ignored us.
Yeah. Yeah. It was.
Yeah, you guys were.
Yeah, we were in the water with
Oh, sorry.
The ROC was circling like sharks around the light. And Ken and I were fine.
Yeah, I think early on we got a few questions, and we really went down into the weeds with the technical details, and then they just stopped asking questions after that.
Although we also triggered that question about commercialization of these chips, and what are the barriers to it actually being used by end users. And they felt that, and they've repeated a couple of times, that the cost per fish is too high. Because if you look at it, if you're doing it, you know, on a small number of fish, you're looking at probably $10 to $20 per fish. Now you're getting a lot of data, 56 genes type thing, but still the question was benefit versus cost. And the benefit is, and since this is a GEN-FISH meeting, I wouldn't tell this to Genome Canada. I think one of the major failures of Activity 3 and 4 was the lack of the synthesis. And most of the publications that are coming out tend to be sort of looking at gene response at a gene by gene or a PCA, but looking at bigger patterns is what we really need to do, so that people who don't know qPCR from a hole in the ground can still use that data to answer their question. What's coming down the pipe in my Atlantic salmon in Normandale hatchery? These genes have started ticking upwards, and it's these ones, but not these. What does that mean? Okay, there's some kind of parasite coming in, and we're starting to see a response. We're not there yet, and I'd hope we would be. But I think we have the data to do that.
And potentially Analisa's random forest, like reinforced learning, machine learning stuff, I think that could help with it as well.
Yeah.
So I want my data point.
That, another general observation, but I think it wasn't part of the original plans. I recall in the grant that this was necessarily meant to be applicable to non-lethal sampling. Never. And that idea kind of got crept in the first 2 years. And when you realize the limitations of the open array, and then you've got this extra incentive to make this applicable for non-lethal sampling, I think that you've got two goals that are not necessarily mutually going in the same direction. If you were to start from scratch to design this for non-lethal sampling, you wouldn't have necessarily started where we started. And that's, I think, also important to keep in mind moving forward. Well, to answer that, when that first came up, non-lethal sampling, I was thinking of a gill biopsy or muscle biopsy, and I thought that would be fine. But then we started getting to mucus sampling and, Lord forbid, blood and environmental RNA. And I was sure that environmental RNA was a total waste of time. And, Shahin's work at Yellow Island Aquaculture, out of the 24 genes, he got 9 that were actually producing reliable signal with environmental RNA. So a water sample that was holding the fish. He then did the same thing with the Freshwater Fisheries Society. I'm not going to go into this detail tomorrow, but there they had, I think he had 9 fish in a tank. At the Freshwater Fisheries Society, they had 10,000 fish in a tank that had been there since they were ponded. And he said he's seeing a much higher success rate in terms of eRNA signal or transcriptional signal on taking a water sample. When we first started using the open array, I didn't think it'd be sensitive enough to do even a mucus swab, much less environmental RNA. But it seems to be working, and working reliably in a way that is surprising. But you're absolutely right. I would have considered going a very different way, maybe a different platform, if we'd started out saying we wanted to do environmental RNA or a mucus swab, so non-invasive. Because really, in the end, it depends somewhat who's your end user. In a fish culture facility where you've got tens of thousands of fish, killing 10 fish or 20 fish or 30 fish, it doesn't matter. And your list of immune genes, you measure them in the spleen, in the head kidney. If you've got them in immune organs, wouldn't pick other non-lethal tissues because it doesn't make any sense if you want bang for your buck. That's a whole other thing, but important yet to keep in mind.
Yeah, and I think, because that was an add-on to what our original plan was, again, we didn't do it in necessarily the best possible way, but we now have the transcriptome of the mucus RNA from the bull trout's work that Analisa talked about, and so then we could figure out what are going to be the good markers representative of temperature response, and then then design chips based off of stuff like that. And so we could have gone with that approach if the goal at the beginning was non-invasive sampling methods for quantifying various stressors.
That part of the issue is that GEN-FISH had a very broad end user base.
And we had to respect that we pitched both fish culture as well as fisheries management, as well as fish conservation. And those three are the tough ones, right? And I did not actually really believe that fish management would find a use for the STP chips, just because the fish are out there in the lake or the river, and capturing them is going to be a problem. And what information are you going to get until they're all half dead? But then, you know, Chris Wilson is looking into using it as a monitoring system. So we had to choose a path that provided the biggest bang for as broad an end user group as possible.
Call actually seem to emphasize sort of aquaculture, economically important fish more than, I mean, I think like of those three, conservation was the least.
Oh yeah, conservation, we snuck that in.
Yeah, yeah.
What the primary thing was fisheries.
Yes, okay, fisheries.
And you may remember Steve Cook nearly blowing a blood vessel when Genome Canada said, well, freshwater fisheries aren't real, are they? They're not worth anything. The recreational fisheries aren't really a fishery. Because we were arguing that recreational fisheries are worth, you know, millions of dollars in Canada. And one of the Genome Canada people said, yeah, but that's not really a fishery. So yeah, that the conservation angle was put in because we wanted that.
Yeah.
So the recreational fishery is 3 times.
Oh yeah. Not according to Genome Canada.
It's not even close.
Yeah. Well, the recreational fishery at Great Lakes compared to commercial, it's 10 times higher. So. Yes. Yeah. Now you're attacking my lakes.
On a much smaller scale, but it's the same trend in Manitoba.
Yeah.
It's shocking that that wasn't on the lines. You know, Canada, the importance of the recreation.
I think the call for the LSARP that we proposed or awarded was targeting marine species. I don't think it ever entered their mind that freshwater would be the focus of one.
Steve has a whole paper on invisible fisheries, invisible freshwater fisheries.
Yeah, the collapse.
How would you have done things differently if it was for marine?
Well, interestingly enough, I would not have proposed this for marine fish. I think that marine fish have a whole myriad of additional complications for environmental DNA. You know, Matthew and I have discussed that. And then when you start talking about capturing marine fish to do gene expression, that's a nightmare by itself. And then species diversity. Again, what species do you do? What stocks do you do? It would be a very different project if we targeted marine.
Mm-hmm.
And it's actually kind of remarkable we weren't dumb enough to say marine and freshwater.
Snuck in anadromous, does that count?
We sort of had to, because those were the fish that people cared about.
What's your favorite fish organ?
Organ?
Organ.
Gill.
Yeah, not, because we did brain, but so thank you. Dennis, Finn and I used the brain shed with Chris Wilson too, we saw it. But do you think if you have to push the tissue and there's no invasiveness involved, like that wasn't a concern?
Well, I like the gill because you can get a lot of RNA for very little tissue size. It can be sampled non-lethally, despite, you know, people still have trouble believing it, even though we have heaps of data that show you can take a gill sample, put a tag in a fish, chuck them out, and they live and they're fine. And then it homogenizes really well in the lab, and it just works really well for a variety of different types of responses. So that's always been my personal favorite.
He's thought about this before.
I think people ask me why I keep looking at the gill.
Okay. He's wrong. It's liver. That's my view. Good.
Ken's convinced me of how well gills work. The mucus presentation this morning could be a game changer for getting uptake for our lesions.
Yeah, that's great to hear. Thanks, Chris.
Yeah. Okay, how are we doing time-wise? Yeah, okay, thank you. You know. And
So the folks who are actually responsible for doing such an amazing job with Activity 5, unfortunately, aren't here today, with the exception of, is Steve still online? That'd be great. So it's Dr. Amy Fitzgerald from the University of Windsor. It's Dr. John Levernois from the University of Guelph and Dr. Tongji Lee from the University of Guelph. I'll be talking a lot more tomorrow and digging into what their results were. So if I say anything confusing, that's on me. It's not on them. And I'd feel really bad if I'm not going to be critiquing any of the work they did. They're not here to explain themselves. And there's nothing really to critique in my mind. In terms of like, what would I do differently? I can't say necessarily. It's up for them to say. So I just want to put that caveat out there. But I can talk about what the deliverables were for Activity 5, and that was to combine all the information across the different activities, including the research that emerged from Activity 5, to be able to produce a decision-making toolkit. And we are about, I'm going to say 85% there in terms of the FISH survey toolkit, which is a decision-making toolkit, whether or not to adopt eDNA alongside or instead of conventional sampling. And that was put together by a research assistant working with Dr. Levernois. It still goes out, it's going out to our co-producers. It had been pilot tested. Again, I'll talk to you about it tomorrow a little bit more. It's already up on the GEN-FISH website, but under a password protection, just so that right now our co-producers can take a look at it just yet. And our fish health survey or decision-making toolkit, it's still in its infancy. We're still trying to figure out what are some of the really important variables that our co-producers have been telling us that would be important to consider. But therein lies the problem, like what Daniel said, is because the knowledge about what this STP chip, when there's a GLFC STP, which is the Science Transfer Program. So again, we're like sharing acronyms, again, what that actually means. So you don't know what you don't know. So what would happen first, what I think we still need to kind of establish is this outreach, this connection with folks so they can become familiar with it, to be able to ask relevant questions, rather than the first question being, what is transcriptomics? And I know that we did have a GLFC Science Transfer Program grant taking a look at omics in general for fisheries management conservation. So those who are more involved in it, please correct me if I'm getting this wrong. And it was to bring omics in general to fisheries managers. I think that was our target audience. And again, we're following up on that. So if anything, we're the activity that's the most behind, which was pointed out to us by the ROC. And again, I don't think it's any fault through any of us. I know it's going to sound like excuses, but I'm just really, really proud of what the Activity 5 members did. And across all the different activities, COVID slowed everything down. So if it slowed everything down, we're already talking about how far eDNA has come since 2018 and the legalities that were first raised. I still remember talking to Nick during the development of the proposal, saying, okay, what kind of these, because GELS, which is my activity, stands for the genomics and the ethical, economic, environmental, and legal implications. And so we thought, okay, what's the L in here? And just talking to Nick about whether or not these results are legally defensible. And Nick, please chime in if you're still online. And it's something that our Activity 5 members didn't really highlight as being an issue. Steve, I know you're there, and maybe through all your research with Vivian, you can comment on that. But based on what Amy looked at, or what Tongji, and even what through the end user committees, the legality wasn't something that I'd really heard. So that was again interesting thing. So my point is, with this kind of evolution of the acceptance of eDNA, you could see it change throughout the lifetime. And so with COVID and the evolution of it, by the time all this new updated information came, it really was towards the end of the grant through no fault of any of the activity members. And then with the transcriptomics, that was really highlighted, not in terms of the delays or anything, but again, like talking to our co-producers about what it is. A lot of that information was filtered to us through word of mouth, through the connections, let's say that papers that are all across all of our activities, and I'm going to go into those tomorrow, including our partners, what emerged from the publications we did and the studies we looked at, emerging technologies. We have papers on co-production. And so that really filtered into activity 5's transcriptomic toolkit, which basically wasn't a lot. And so it was actually reflected in some of the papers that we produced. It wasn't really discussed, thought of, and in terms of adopting, it's not on anyone's radars yet, because the evolution of that, we're in its infancy. So where we are now with transcriptomics, I would say it's the equivalent of eDNA mid-2010s, if not sooner. So I'll open the floor to any questions you have in terms of perspectives, findings, and again, I can go through it now or more, but I want to make sure I have Steve's points being expressed as well as any comments.
Want me to layer in a few things right now?
Yeah, sure. Go for it, Steve.
Cool. So awesome. Thanks, Tina. That was perfect way to start things off. I think the L in gels is the only one that we didn't really dig into, L for legal, but I don't think we needed to. You know, you think about some of the other topics that are pursued in the context of Genome Canada projects, and some of them inherently have legal implications. It's about setting regulatory guidelines or drug discovery and so on. Ours is really about providing managers with tools to do their jobs better. So yeah, the legal, you know, I'm sure if we, you know, dug deep enough, there's some legal stuff, and maybe, you know, expert witnesses will rely on data we use, but that's kind of neither here nor there. It didn't justify a dedicated research program. That said, we covered every other letter in that and covered it well. And I think, having been involved in some of the other gels, oftentimes there's a single token social scientist that gets $10,000 thrown at them. We had a team that was diverse, that spanned a variety of disciplines, and that allowed us to really hammer on all sorts of different angles and perspective, doing very different things. And with that, of course, there's this end product or products, these toolkits, but there's a lot of the foundational work that we did on the front end that is benefiting us on the back end. We had to wait to have something to share, so we need the other pieces, but we still built a foundation, and there were missing pieces. We couldn't just push a toolkit out there. We needed to think about the receptivity of the community in different spaces, and who is the community? That's, you know, there are multiple groups, whether it be Indigenous governments and communities, whether it be anglers, whether it be folks that work in parliamentary government as environmental managers, consultants, and so on. So we did that work, and I think that work will pay dividends that will extend well beyond GEN-FISH, because some of that work is more germane. It's about technology and new ways of doing things, new ways of generating understanding, and realizing that new, even if better, doesn't mean that it will be embraced. There's all sorts of reasons why that's not the case, why there's challenges. And we really picked away at understanding that. And by understanding it, it helps you to try and overcome those barriers and challenges. So a lot of our early pieces were thought pieces We didn't have any data to work with, and it was COVID. And so we just started, oh, it'd be fun to write about this or that. I think it would be fun to revisit some of those, Tina. For example, the one that you led with a number of our end users about the potential for this technology to interface with their day jobs. I think it'd be cool in 3 or 4 years. This is after the funding is very much evaporated, but to revisit that and look at the extent to which that was actually realized. So from the promise to the reality. And so just as I think about the gels, I think we're going to have some work that continues long after. I think there'll be opportunities to come back and do social science work to look at the real uptake. We'll be able to do some really basic stuff in terms of downloads and, we get, simple surveys linked to folks that use things. But I think in the longer term, again, 2-3 years later with some imaginary funding we'll have to find, I think that's where there'll be real opportunity. And doing that will allow us to feedback and really do things better next time. We just have to figure out who's leading the next time grant. So
that's great, Steve. Yeah, thank you. Perfect. And now I would just get to that comment first. Is that okay? Nice. So they're setting the groundwork. Yeah. Margaret.
Yeah, and actually what I was going to say pretty much follows from that. So we didn't explicitly, consider whether, the legal sort of reliability, but everything that we're doing sort of contributes to that. There was a paper published by a US-based group within the past few years that was looking at, basically is eDNA ready to be used in a court of law, and they were evaluating whether it met what's called the Daubert standard in the U.S. It's a legal principle used in U.S. federal courts to determine the admissibility of expert witness testimony, especially scientific evidence. And I think there's 5 criteria that scientific evidence has to meet to be ready to be used in a court of law. And they concluded that eDNA does sort of meet that. And so what we're doing, not for eDNA in general, but for any particular assay, or you could maybe extrapolate to the fish health work as well, that making sure that any given assay or if any given health chip is sufficiently reliable, that experts could use it in defend it.
Yeah, and that just made me think that we're not even there yet for using transcriptomic data to show, let's say, this fish population. Can you imagine that you're like, this fish population is in threat? There's a threat of it becoming like we haven't diagnosed it, but we've prognosticated that, oh, this is gonna be sick. Like, well, that's not. Try to defend that.
Yes, and
it's gonna be tough.
Or
yeah.
I think that what we're doing is contributing to that. And the first step isn't the legal aspect,
should be the last.
The more immediate end users, like first trying to satisfy people who might want to apply it for making conservation and management decisions. Are they satisfied that it's meaningful? Legal is down the road.
Yeah. So I'm going to turn this back to activity 4 and 3 members. How reliable are the chips? Do we know that yet? And showing there's different aspects of where reliability or validity lies in that pipeline and that chain, but in terms of showing an effect, how confident would you say if you're put on the stand, Ken? You're like, your future funding depends on this.
Take the pick. Was it 100% confident?
Yeah, true. I know.
I think honestly they do a very good job of identifying when stressors start to hit the fish and it starts to impact them.
Yeah.
There would be other ways that you could do that, but I think if you want to just see at the cellular level where a stressor is going to have an impact, they do a very good job of showing that.
Yeah. I agree.
Part of the. Go ahead, Steve.
So Nick has had his hand up for a bit, but I think he's going to take us in a different direction, maybe. So I'm going to just add one thing onto what Ken just said. So think about cortisol. It's something that's been measured, and there are hundreds of studies on it over the last, 30, 40 years. Would I feel comfortable sitting on a stand being questioned about, with certainty, can you, will you attest that? And it's just so, well, jeepers. No, this stuff's complicated. And so we're always after the silver bullet biomarker, and I'm not sure it exists. What I like about the omics approaches is that you analyze a number of things, and they collectively point through different systems towards a more cohesive understanding than just 2 or 3 biomarkers. And so I think that systems level understanding that's rooted in biological function, I think, has a lot of promise. And so there will always be uncertainty with these things. We don't know what happened to that fish before we sampled it, if it's a wild fish. I really like how it's not one thing. We heard in one of the presentations this morning about being able, the random forest being able to assign fish to different groups with a level of certainty. I believe it was 90% assignment success. That's a number that a judge can wrap their head around, right? And realize, we don't have certainty very often, but you can tell them about where the level of uncertainty, which I think is helpful.
So, Nick, you want to unmute yourself?
Nick, go ahead.
Yeah, well, you sort of indicated that I brought up this legal thing. It's because I thought the acronym sounded better with the L in than out. But seriously, what I was thinking at the time when I brought that up was, with my history of working at DFO, is that managers make decisions that have to be legally defensible, right? And the best way to be legally defensible is to be scientifically defensible. As simple as that, right? And so, I think GEN-FISH has contributed a lot to making the use of environmental DNA scientifically defensible. So we've implicitly moved the yardstick on the legality, I think. And probably made managers more comfortable with using environmental DNA as a result, not just because now they believe it more, they believe it's actually providing real evidence, but also they know that there is science to back up their decisions when making decisions based on environmental DNA.
So yeah, go ahead.
So one of the things that I've found interesting with the eDNA was early on, we identified potential ethical issues. Because whether you like it or not, when you've got eDNA, you don't just have fish eDNA. And I don't know if those of you on the attended the ROC, we brought that up at one point and essentially was totally dismissed as being almost silly. With the advent of wastewater surveillance, now there are a bunch of opinion paper articles out there about what are we going to do about these samples that have human DNA in it that nobody yet has pointed out. But it's not just the the COVID virus that they're picking up. There are microsatellite markers in there. You could identify at least the race of the individual humans that contributed to the waste, but possibly even individuals. And nobody's thought about this. It's just sort of eDNA. Nobody's sort of thought, okay, it doesn't just come off fish.
Yeah, that did emerge in Amy's focus groups and some of the sectors that she spoke with, and to the extent of it, again, maybe around some legalities, but it was just more in the lines of ethics. You're holding a container that contains personal information potentially. Privacy. Yeah, but nothing that it was just raised. And then the question comes to us, like, what's our responsibility as researchers to collect it? Do we also need a research ethics board certification that we're going to be collecting human data? But as long as we don't use it, but then if it becomes archived, like there's going to be some really interesting questions that emerge from that. And who then can give permission for it to be used? That didn't really come up. Margaret, you have an answer for that? Oh.
All the ethic issues have been the last 2 or 3 years.
For some reason, I thought the ROC had raised that, or sort of had scoffed at it more recently than 2020. Maybe I'm misremembering.
And maybe I'm big blur in my mind. So.
Oh, sorry. Go ahead, Steve. Sorry.
So one of the really interesting findings I thought that came from Andrew Howarth's survey or interviews of fisheries managers in Canada was around the so-called supporting cast. And you always think about fisheries management being done by fisheries managers. And he really did a good job of mapping out the roles, which varied, from case to case and region to region and so on, of all of the other actors in that space. And it's immense. And it goes from full on, really well done co-management where Indigenous communities are exercising their sovereign rights through to, in Ontario, there's fisheries management zone councils that are populated by relevant stakeholders and rights holders that work collaboratively to come up with fisheries management targets and then try to hold government to account to do that and so on. My point is that when we think about who we're selling these ideas to, it isn't just the fisheries managers, right? We really do have to think about messaging this to that broader ecosystem of actors. If we only, and I think, I probably knew that if I thought about it hard enough, but we always assume, we're making a management toolbox. Who's it for? The managers. Who are the managers? MNR, like people that work for parliamentary government natural resource management agencies. And I think that's changing in a good way, but it also complicates things for us in terms of who we message and how we message. I think it'll be interesting to reflect back in a few years to see if we did well enough in that respect. If it's the kind of thing where the managers all buy in, but that supporting cast that gives it the finger or is suspicious of it, then that tells us maybe we need to do more work in this space or that space. That said, I think we did dabble in all of those, from Tongji's survey work targeting rec fishers to some of the community conversations that Amy had and so on. We did some of that, but of course you always wonder, is it enough?
Mm-hmm. And to add to that, Steve, thanks for bringing that up. Tongji's, she's a behavioral economist, so to see how people can be nudged, any kind of behavioral economic nudges to get someone to adopt or to be willing to give up or to accept anything. And she said one way to accept novel technologies is to have early adopters. And typically the only early adopters, and Steve, correct me, are the ones that have this research capacity, which typically falls on government. So it's going to be interesting whether or not, if government adopts it, is it going to be a domino effect? Or like Steve, you said, will it be, the players that will have to adopt it in its stead, especially not with the eDNA, because that's already almost like a done deal. There's no need to convince folks to adopt eDNA. But it's this transcriptomic work that I think might be a tougher sell because it's more novel at this point than eDNA. And so how do we get someone to uptake it? People have to have that capacity. And so with transcriptomics, we still haven't produced something that allows it to be a let's set up a lab. Great, you could send the samples to, but then how do we analyze the data? What are the data actually telling us? And then if we put something in action, did it work? So I don't think we're there just yet, but it'd be interesting to see who were the early adopters of this technology after academics, and how quickly did it spread to being adopted by all the other sectors and users. So still more work. So that's GEN-FISH 8.0 maybe, you know.
And as you touched on, it's, you know, it's going to be people, I don't know if Chris Wilson's still on here, but people that work for natural resource management agencies, but with a science hat on.
Yeah.
That also, you know, a bunch of the social science work that we've done adjacent to this really shows us that it's, you know, when science is institutionalized, that's when it really sort of validates it and leads to widespread internal adoption. You know, science, like for better or for worse, science done by a DFO scientist will always be viewed more favorably than by a manager at DFO, but then by science done by academics or anyone else. It's just, you know, hey, we're on the same team. You understand exactly what I need. There's a trust thing there that's just, you know, really difficult to get around. So when you have people that are, whether they're early adopters or internal experts that are saying, yeah, I've done my own ground truthing, and yeah, I think this is a solid tool, you know, with these caveats, that's when I think the adoption really happens. We as academics can wave all we want. If you don't have an internal champion or expert that's able to again, institutionalize it, I think that's way more challenging.
Any questions, comments, feedback? Yeah.
The original sort of pattern that was described by Genome Canada, actually Ontario Genome Institute, when we were developing the idea of an LSAR proposal, was that there was the sort of the natural science, and then the GELS was meant to almost be the overseer. And that the GELS group was supposed to say, hey, watch out, this is going to be a problem in terms of adoption. And then you let the natural scientists say, okay, we're going to change the direction in order to avoid that barrier for adoption. And I think one of the sad things about the impact on COVID is I think that the time for that feedback loop was lost because the lazy natural scientists didn't get anything done for 3 years because they couldn't get in the labs. They couldn't get the labs to get information that could be evaluated. And so the activity 5 folks pivoted, and as Steve was saying, developed opinion papers that framed the issue, and then, you know, started looking for initial ways to evaluate this, but it couldn't be specific.
Yeah.
And
by the time that information came out, like, you're already set in terms of this was our protocol. This is what we're going to do. So there was not that kind of dynamic like you're talking about, like back and forth. It only really started when, and also because the projects that were pitched, we said we'd start the end user committee in the very beginning, I think it was, to provide that feedback, but it was called the end user, but our co-producers, but there wasn't really anything to discuss, and there wasn't a structure. So in 2020 hindsight, you know, we're talking about eDNA, if we had that standardized protocol from the beginning, if we had started with our end users from the very beginning and say, okay, what are some of the questions you want us to answer, but to keep it in check. Our whole proposal was based, though, on all the relationships and the networks that we had made with what are the types of questions you want us to answer. It was whether or not we kept on track. But I think we did, because we received so much feedback. And then you can see how many spin-offs that was done. It didn't need to rely on activity 5, because Amy was there doing focus groups and media sweeps, and that was her own forte. That was her own wheelhouse. Tongshi was doing her behavioral economics, and again, what was emerging from there was just reinforcing what was already known. So I think that our end users and through our HQP really helped refine everything. And so I'm okay with the lack of activity 5 members per se, I'm talking about like the 3 ones that were listed. I'm not talking about any of the work that Steve's done, because I did feedback and everything, or what Thais Bernos, Dr. Bernos did with Nick's group and all other co-producers. All of that went into the mix. It all went into the mix. We just formalized it sometimes in papers, or we had it informally communicated to activities 1 through 4. So, you know, I think we did, just not in the way we had thought of.
Yeah. I also think our GELS team included crossover folks. So Tina, myself, Nick, all are, you know, NSERC people first and foremost. And we dabble and collaborate and kind of, you know, span that gap. And I think that's helpful in terms of it not being an us or them, you know, like those, you know, the economists and social scientists that none of us know are over in that corner. And so there were a few of us that were sort of boundary spanning in that sense. And I think that helped to ensure that what was being done there, that there was connection, and, you know, we knew just enough about both to be helpful and lean as we needed in both directions. So, and I've seen it go the other way, where there was lack of a common language. Right. And that crossover was missing. And so even look who's here today presenting on behalf. It's somebody, Tina, who's one of those boundary spanners. And I think that's super helpful for getting buy-in from the science community, but also ground truthing it from the beginning to the best extent possible. I will also remind everybody that right from the very first time we met with The Rock, we were getting hammered on co-production. We wrote that paper to some extent to say, we actually know what this is, by the way. And yeah, so there, and the idea of, you know, oh, well, we want you to co-produce it. And it was, again, during a pandemic. And by co-production, they meant working hand in hand with Indigenous communities to define the questions. But that is not a fast process by any means. It's an important process, but for that to be done, that needed to be done on the front end. So again, we relied on existing relationships that were sufficiently mature and sufficient trust that we could build from there. So yeah, there again in hindsight, but that hindsight is partly about the structure of the grant, right? You have to write it. Here's the timeline. Here it is. You know, you need to lock in what you're going to do and convince people. And then you get the money and they're like, oh, wouldn't it have been nice if you did this? And you're like, well, we did not have the money to do that, you know, 18 months ago. So if anything, I would love to be able to provide feedback to Genome Canada about having, you know, relationship building money for the front end. And then I've already stated my interest in revisiting what we've done 2, 3, 4 years later. And so having pots of funding that would enable that as well. I think that would be a really nice way to bookend it properly, instead of this. The bookends are a little bit too tight, if you will. I'd love to see them extended in both directions, for the planning phase and for the wrap up evaluation phase.
Mm-hmm. We're happy?
Yeah, well, on Steve's point, a little bit of ancient history. GEN-FISH started out a really long time ago as a nascent discovery accelerator proposal. And the Discovery Accelerator program gave us $20,000 to develop relationships before we actually even put in a proposal. So this was, we hadn't even been funded, but we didn't get funded, but we had fun spending the $20,000.
Yeah.
So
yeah,
it's an interesting point that Genome Canada doesn't do that.
Yeah. But they expected it of us.
The relationships to pop up in 4 years.
Yeah, just wasn't realistic. But we did forge new partnerships.
Yeah.
Like, think about all the new partners that joined GEN-FISH after the fact. Like, some of them we couldn't develop because of COVID. Some of them just drifted. But I think we gained more than I think that we lost. But I'm looking at the page. You might have the final numbers on that. Because I think as our networks grew and grew because we all networked together. And so just synergistically, we had more partners and bringing others into the fold, it did grow and word of mouth. Now Ken's talking about, and you're like non-GEN-FISH members hearing even through conferences and approaching you saying, hey, look, this is really cool. Having said that, we also have end user committee. And at the last meeting, Steve can talk to this, we asked about, okay, how do we make our products more well known and distributed beyond just our typical sectors? And they gave us some really good pathways and avenues to pursue to come talk to. Some of it was in the Great Lakes, I think, Fisheries Indigenous Network. Some of it is through LinkedIn, through our environmental consultants, and I think there were some others as well that we can try to target, not just through the standard academic routes. Yeah, Margaret.
Well, it's not like the other activities are all just pure research, and then it all falls entirely on activity 5 to make sure that there is uptake at the end. But I think, yeah, that I think that what we wanted to accomplish, it's accomplished, regardless of what activity it fell in.
Agree. Because it always felt activity 5, like you're not doing enough co-production. And activity 5 is like, we are not, it's the natural scientists should be the ones co-producing with the partners. We can't, you know. Yeah, exactly. Yeah.
Side, to have conflict or falling out, had nothing to support.
Yeah, there was no infighting with the natural scientists, and there's no conflict between natural and social scientists, or even, you know, some of our community members. We just listened to what they wanted, and they didn't want us, we walked away. That's something we still need, science transfer.
That door, I'm sure, is still open. It wouldn't have to have been done within the time frame of GEN-FISH.
Yeah, we did say we were going to have the very, very last quarter, I think until end of June, we're supposed to get this translational product done. But I don't think that's going to happen. So, you know, that's too late now, ROC. We got the money.
Yep.
Good. Well, stay tuned for tomorrow when I present more details. We can have a more informed discussion when you know more.
Question.
Sure.
So when you think of John, Tony, and Amy, you think they had fun?
Of course I did. Yeah, I'll speak on their behalf for sure. John never, he was adamant saying, I don't know what multi-criteria, MCMD or multi-criteria decision-making analysis is. So he hired someone from physics to compose it. So they both learned about MCDMAs. And I know that Amy really enjoyed her time working with Chippewas of Nawash First Nations group. And she was invited to the community feast. So she was really happy about that. And Tongji, I think that she, unfortunately, I think she got hammered the most because it was like, where was her Indigenous involvement, engagement? Because she's like, I'm a social scientist and my sample size, my participants are the public. And so she makes calls out to the public to participate, except for when she did like, I think it was the recreational fishers. I think she did that too as a group. But again, the ROC was like, why aren't all your social scientists working with Indigenous First Nations? And Tongji hadn't had established any. And then she always said, it's not the nature of my behavioral experiments either. But so that was kind of like, okay, not that she was against it at all. So but I still think, you know, she had fun doing it. And Steve had a blast. This is all the time. Yeah. Any other questions? We're good? All right, good. Thanks, everybody.
So this is the wrap up before we all go back and relax before the feast tonight. The things that I was planning on saying have been said multiple times today by support personnel, by HQP, by PIs, and that is, you know, it's really easy to look back on a project like this and think it should have done this, or it's really too bad this didn't happen. But all of us at the end of the day come to the conclusion that we accomplished something really remarkable. And the original proposal was very ambitious. I think Margaret said insane, and it might have been that. But I would say we got within 80% of all of the targets across all of the different activities and the objectives within the activities, which is remarkable. And, you know, I'm aware of other Genome Canada projects that were defined so narrowly that I'm amazed they were funded. Sequencing the Atlantic salmon genome was a Genome Canada project. That was the entire project. And it was funded to the tune of close to $5 million. And they did not complete the project in the time that they had. So they actually did the sequencing, no problem, but they couldn't annotate it. And I'm not faulting them at all. They were dealing with a very different technology. We could sequence and annotate the Atlantic salmon genome in a month and a half now. But the point is that we shot for the stars and we damn near made it. And where we got has produced a bunch of information that will outlast GEN-FISH by decades. And we've actually produced tools that I think people will be excited to continue to use after GEN-FISH is done. And, you know, so I questioned Margaret about those species we didn't get the eDNA assays for. Are they important? I suspect that Margaret's lab might work on some of them, and I suspect other labs might keep working on it without any funding, maybe on NSERC funding. And this is to me, the most rewarding part, is that we generated combinations of researchers, we trained HQP, and we accomplished a lot. And I think we should all pat ourselves on the back, and all the HQP who've graduated and moved on should be very proud of their membership in the GEN-FISH family. So I think we did a good job today of looking back at what we've accomplished. And I think hopefully we're all very proud. I know I am. And tomorrow I'm even more excited because we're going to look at new directions and into the future about what GEN-FISH can do, sort of the legacy part of the AGM. Would anybody like to say anything? Any comments? Or are we done? We're all good? Thanks.